You take it away.
Thank you. So, good morning, everyone. I want to welcome participants to this session that will focus on the discussion of capacity building for DSI. So, some of the options available, and also we also will have the opinions from the panelists. Okay.
And I'm Amber Schultz. Good morning and welcome on behalf of the DSI Scientific Network. We're really looking forward to having you here and to bringing some new ideas on capacity building into the discussion.
Okay, thank you, Amber. So, dear delegates, observers, and stakeholders, good morning and welcome to this session, number 6 of the 5th Science Policy Forum for Biodiversity and the 8th International Conference of Cerebral Science. I will first introduce myself. I'm Charlie Mabala. I'm part of the Gabon delegation. I received a Bachelor of Science in biological science from the University of Nebraska in Omaha, then a Master in Biotechnology from the University of Nottingham Trent in the UK. And I completed my doctorate in environmental biotechnology from the University of Supergro in France, in Montpellier. So, but before we start, I would like to thank the organizer. I know it has been a little bit hectic this morning. We've tried to figure out in which rooms we have to be. So hopefully everyone is here, and then we will continue without any other movement. So I would like to thank the organizer to allow us to be here. So especially I wanted to thank the International Union of Biological Science, the German Research Foundation, and the ABS Capacity Development Initiative. And also I want to extend my warm welcome to all of you who are following us online. So I would like to present first and foremost the DSI Scientific Network, which is a global network of 70 scientists that comes from 25 countries, formed in 2020. And then they formed in order to give a platform to the scientific community to discuss the global management of DSI at the political level. So, and then the mission of the network is to contribute to policymakers and stakeholders, contributing to the understanding of DSI, its application, and its contribution to research, to biodiversity conservation, and to public health. So we had prior to this one, we had a side event on December 8th that were really discussing on the policy option of DSI and the management within the CBD framework. And today's event, it's quite interesting because it will focus on the discussion on the need. It's an open discussion on the need of DSI capacity building that will enable transparent, equitable, and certainly use and access of DSI worldwide based. So after that, I will introduce the speakers. So the presentation will be a 7-minute presentation, and with 3 minutes of exchange. So the first presenter will be Melania Munoz Garcia from the Leibniz Institute in Germany. Melania Munoz Garcia holds a master's degree in biology from University of Costa Rica. And from 2013 to June 2022, she worked at the Costa Rican governance, Ministry of Environment and Energy as the ABS national focal point. Since July 2020, she has been working as an ABS project manager at the Leibniz Institute. She will present DSI and capacity building and just an overview. Our second presenter today will be Anne Marcottney from the Genomics Institute and the Native Biodata Consortium from the USA. And Marcottney obtained a PhD from the Dublin City University and has since carried out a postdoctoral she had carried postdoctoral research fellowship in Manaki Union for Genomics at Tora, and at the National Human Genome Research Institute. She acts as the director of a European Reference Genome Atlas, the pilot project, and the director of partnership and strategy for my Afro Data Biobank. She will present the GBF is connected to DSI from barcoding to eDNA to the whole genome. Our third presenter today will be Tamiro Oljira, from the Ethiopian Biotechnic Institute. Tamiro obtained his Master of Science in Biology at Addis Ababa University and obtained his PG from the Department of Molecular, Cellular, and Microbiological Biology at Addis Ababa University as well. From 214 and 218, he has worked as an associate professor at Aramaya University. And for 2018, is a senior, since 2018, is a senior researcher and director of the Ethiopian Institute of Biotechnology. So he will present today, Understanding Human Genetic Diversity from Improved Health. And finally, our fourth presenter today will be Suwel El Janabi, from the ABS Capacity Development Initiative. Suwel El Janabi studied geography, ecology, and economics in Bonn, in Germany. And Suwel Janabi is today in charge of conducting several multi-stakeholder processes in Africa and the Caribbean in close working relations with the private sector and the donors at the ABS initiative in Norway, Denmark, European Commission, France, and the German Federal Ministry for Economic Cooperation and Development. Today we'll present lesson learned from ABS capacity building. So you introduce yourself?
No, it's good. That's it.
Okay. So we start with the first presentation.
Thank you, Charlie. Can you hear me? No. Now, can you hear me? Yeah, better. Thank you, Charlie, for your introduction. I will give you an overview today on how DSI can contribute to bioeconomy and some ideas on capacity building, highlighting the importance of the implementation of the DSI's policy option number 4 that is on the debate in this COP. First, let's take a look on the market. What is the estimated value of the global biotechnology market? By 2020, it was estimated in more than 600 billion US dollars, but this market is still growing. And by 2026, we expect a market of more than 1,000 billion US dollars, being the pharmaceuticals the biggest section of this. And also take a look at how is the global biotechnology market distributed. As we can see here, the global north dominates the market and the global south has just a little piece of the cake. So the most important question during this discussion today will be try to answer the question, how can capacity building on DSI improve this unevenness? Let's take a look at the bioeconomy policies around the world, understanding bioeconomy as global growth based on bioproducts and bioprocesses, reducing the impact on the environment. And here you can see the dark green countries have specific bioeconomy strategies, and the light greens have some strategy in their development plans that are related with bioeconomy. But here you can see also the unevenness between or among the global south and the global north, where in the global north, you can see that there are less plans. As an example, this is how the national bioeconomy strategy in Costa Rica is done. First, and in the bottom, you can see that it includes obviously agriculture and reduce the impacts on agriculture in the environment, also biorefinery. But in the top, you can see how biotechnology in all their different fields also is an important part of bioeconomy in Costa Rica. And of course, for the Costa Rican government was very important to include a section on access and benefit sharing. And I would like to highlight that now Costa Rica has a virtual platform to automatize the systematization of non-monetary benefit sharing, taking into account that knowledge production is the first step to create capacity and develop bioeconomy. Another example is Colombia. Colombia also has a economy strategy, and they also highlight the importance of bio-based products from different fields, green chemistry, cosmetics, pharmaceuticals, and also bioprospection and the development of products from different biomolecules. So it's really important to understand that capacity building of DSI is a way to implement bioeconomy strategies, support economic growth based on biodiversity, sustainable use, conservation, and restoration. And it needs a strategy and robust programs and projects. It is not just isolated trainings here and there. And to implement capacity building, we first need to identify the needs of the different countries, and this is just a summary of the side event that the DSI scientific network presented in Geneva this year, where the Ethiopian, the Brazilian, and Algeria's researchers express their needs on that side event. So you can see here that there is a need to training to produce DSI to be able to analyze their own local biodiversity, but also training to the use of DSI to answer specific questions related to conservation, agriculture, culture, health, situ collection, among others. And for that, it is very important to have the capacity to analyze the databases and improve the use of bioinformatic tools. Also, there are necessities on organizational capacities, for example, public and private collaborations, and infrastructure capacity, but not just building labs in developing countries, but also access to international sequences facilities. And once we identify the needs, we can start thinking on the solutions. So those are some ideas about solutions on capacity building. First, create a beta data centers of excellence focused on regional needs and priorities. As we know, the majority of omics related databases are in the north, in the global north, but and besides these are open access platforms. The strongest expertise is where the tools are building. That's why it is important to build these the biodata infrastructure in the south. And one idea is to partner with a global biodata coalition who is working to diversify both funding sources and geographical locations of these databases. Another example is create a scientific matchmaking platform for DSI capacity building where different stakeholders can find opportunities for financing, training, and mentoring. And this is one example developed by the Costa Rican government that is called the Bio Business Platform. And one side effect of this could be the opportunity for monitoring non-monetary benefit sharing on the target 13 of the GBF. Another idea is create a global sequence center for the GBS related research. Because simple and cheap access to DNA sequences for low and middle income countries located scientists could move the needle of the implementation. So basically develop this center to support GBF related goals, targets, and indicators, and their EBIT recommendations and priorities. And the fourth idea is to build new programs or expand existing ones to include omics and bioinformatics. So first, we need to identify what already exists and then what other things need to be developed. And I will give you some examples. First, some examples of programs, examples of successful programs that already include the omics and bioinformatics, and we can reproduce them, let's say. The first one is Cabana. It is a capacity building project for bioinformatics in Latin America, taking into account the training needs of the scientific community and accelerate the use of bioinformatics. They prepare workshops, training, the trainees programs, and also e-learning resources. And they focus specifically in those three areas. And how it is governed is really interesting because they have partners in the global south, where we can see Argentina, Brazil, Costa Rica, Peru, Colombia, joined with the European Union, but financed by the United Kingdom. Another example is H3 Africa, that is focused on human heredity and health in Africa. This is a consortium of the US National Institute of Health, the Wellcome Trust, and the African Academy of Science. And it focuses on empowers African researchers to be able to analyze the genomics and collaborate with the African and global health. Another example is the Periodic Table of Food initiative. It is just doing taking the first steps, and they would like to create a public database on chemical composition and function of food. So it's interesting, but because they focus on metabolomics. And then some examples of programs that already exist, but we would like to see the omics and bioinformatics included in that. First one is SATREPS. It is a program from the Japanese government, and they focus in these specific areas: environment and energy, bioresources, disasters prevention and mitigation, and infection diseases control. And it's a good opportunity. They are financed by the Japanese government, and they perform almost 200 projects in more than 50 countries around the world. And the last example that I would like to share with you is the Global Taxonomy Initiative. They are focusing on capacity building on DNA barcoding for rapid species identification, and collaborating with monitoring and conservation programs in different countries. So to wrap up, this is a possible vision for the implementation of DSI policy option number 4 in order to reduce the gap in the capacities to produce and use DSI as a driver for sustainable development. So created biodata centers, developed scientific matchmaking platforms, developed global sequencing center, and increased the omics and bioinformatic capacity building. And I would like to thank the DSI Scientific Network, especially to Andrew Hofton and Rodrigo Sara and Amber Hallman Scholz for the work compiling this information, and also to the science policy team that is always willing to hard work, but also to have fun. Thank you.
So thank you, Melania, for this interesting presentation. And which give you a good insight about how we need to collaborate, and also how infrastructure and also the need to really have a good scientific cooperation will enable us to contribute to DSI capacity building. So in the interest of time, what we will do, we just go with the presentations, because we have the panelists also that will intervene, and then if at the end of the presentation, if you have a couple of minutes for questions. So we go with the second presentation. Okay.
Okay, thanks very much, and thanks for the kind introduction. You can hear me all right? Perfect. So I'd just like to start with the acknowledgement that we are sitting on the unceded lands of the Mohawk, and I would like to thank them for inviting us and hosting us on their lands. I would also like to acknowledge the words of Charlie Patton, who spoke at yesterday's Nature and Culture Summit. And he had said that we need a fundamental shift in our relationship with nature. He said that we needed to wipe our eyes, wipe our ears, and wipe our minds, and to reevaluate this relationship with nature, and then to clear our throats to voice the change that needs to happen. And I really hope through my talk today I can do some of that justice. So probably most of us are aware at this point that we know vastly little about the biodiversity that we share our planet with. We know just 1.8 million species out of an approximate 20 million species. We need a reliable and actionable corpus of knowledge about all species on the planet. We need to document our species, and this is a task of unparalleled importance. It has taken us 400 years to identify just 1.8 million species. We cannot proceed at this rate. Genomics, decentralized and scalable genomics, can help, and it can help quickly. For instance, if we want to create the complete book or the complete genome of a single species, let's take a vertebrate, for instance, that would cost today about €2,500 and take approximately 3 days at full sequencing capacity. If we scale that to all vertebrates, that would be about €170 million and would take under a year to complete. If we scale that to an estimated 1.5 million species and created a real encyclopedia of knowledge for all of our species, that would take €3,850 million and approximately 15 years to complete. So generating a genomic encyclopedia of all of life, of all of our species, is technically and financially feasible. The challenge here is that it's actually to obtain the sequences of these species, we must first access them. So the biodiversity genomics community is a growing community, and we are relatively new, and we're trying to think about how we would like to grow. And when we're trying to be intentional about our growth, it's so important that we situate ourselves in the broader context of the biodiversity research enterprise at large and realize that we come from a history of extraction where genetic resources have been extracted from the global south to benefit those from the global north. And so as we move forward, we needed to situate ourselves in this colonial legacy and learn from it to move on. So I would like to highlight a quote from Mae Jemison, who's the first Black female astronaut. And her quote was that the future never just happened, it was created. And so when we were building this community of biodiversity genomics, we had to be intentional about how we were creating it, so too to mitigate the manifestation of this colonial legacy. And so to do this, what we really envisaged was that we would generate sequencing data from all species across the planet, and that it would be distributed on this and accessible in a global database where anybody across the world could access this database and utilize those resources fairly and equitably, but also to bring together a network so that scientists could actually collaborate and work together with their diverse perspectives to maximize the utility of these resources and the scientific outcomes for conservation and preservation of species. So we needed an approach that was embedded with justice, equity, diversity, and inclusion. And this model would really be to build local capacity. In 2018, we established the Earth Biogenome Project, which is an umbrella project of affiliated networks across the globe. And these affiliated projects have come together with the audacious mission, a moonshot for biology, to sequence all of life, to really create this encyclopedia of sequences for all of the species we share our planet with. We are growing, and to date, we have 51 affiliated projects across 110 countries. We have over 5,000 participating members. We are only 3 years into the project, and we've already created high-quality reference genomes or books of life for over 1,500 species across the phylogenetic tree of life, and this momentum is growing. One of these affiliated projects I wanted to highlight is the European Reference Genome Atlas, and this project was established in 2021 and is now funded by Horizon Europe. What we were trying to do with ERGA, or the European Reference Genome Atlas, was to sequence everything across Europe. But our fundamental mission is not to develop highly technical algorithms to really complete sequences for all of species, but rather to invest the time from the outset to build a decentralized genomic infrastructure that could support the production of high quality reference genomes from all European countries, but with researchers from all European countries. So we currently have 234 research institutions that span 38 countries. And so this decentralized genomics network for the first 3 years will produce reference genomes for over 500 species, but this will scale as the project moves forward and this decentralized infrastructure is in place. This is part of a larger European initiative called Biodiversity Genomics Europe, and our partner project there is BioScan Europe, with their mission being to generate over 15,000 reference barcodes from 45,000 specimens. So it's important when we think about when we are creating this encyclopedia of knowledge of our species that we provide appropriate contextual information that travels with that data into public repositories such as the INSDC. All of the genomics data that we upload to public data repositories must have appropriate levels of this contextual information. This contextual information is the how, the when, and the where the data has been produced. Historically, biodiversity researchers have not provided comprehensive metadata, and this can be reflected in that just 13% of sequencing data in public repositories such as the INSDC contain country of origin information. Thankfully, in 2021, the INSDC actually updated this policy to mandate the inclusion of country of origin information. Unfortunately, this doesn't provide the level of granularity to ensure that Indigenous peoples and local communities can disclose the provenance of their information. And when we are building these scalable global projects, we want to ensure we're building sustained partnerships with Indigenous peoples and local communities and support them in exercising Indigenous data sovereignty over their genetic resources and data. To do this for the European Reference Genome Atlas, we thought about how we were contextualizing our encyclopedia of knowledge. And to do this, we came up with a metadata collection procedure where we collected 81 validatable fields of contextualized information for each of the species that we were uploading into public repositories. One of the things I would like to highlight is that we are one of the only initiatives that mandate permit upload along with the genomics data. But we are also the first initiative to scale the disclosure of Indigenous rights into that contextualization of the genomics resources. One of the ways that we did this was partnering with the Local Contexts organization and to introduce these traditional knowledge and biocultural labels and notices where Indigenous peoples and local communities could disclose provenance, permissions, and protocols information associated with all of our genomics resources, really operationalizing Indigenous data sovereignty. With that, these actually travel into public data repositories, for instance, the metadata to biosamples and the actual genomics data to the European Nucleotide Archive in this case. So with that, I would just like to thank you all for listening. And if you have any questions or would like to follow up, I have this slide with additional resources available. Thank you.
Thank you, Anne. Thank you for this great presentation, and then gives you also an insight on how having inclusion of the IPLC is really critical and important when it comes to capacity building and when it comes to knowledge sharing and also in terms of collaboration. So thank you for this interesting insight. So we will go to the third presentation. So I will invite Tamiru to present. He will present on understanding human genetics diversity for improved health. Over to you.
Okay, thank you for introducing me properly. My name is Tamru Oljira, and my institute is Bio and Emerging Technology Institute. There is a name change somehow from what you have introduced. Recently, we have merged the Biotechnology Center with another center, an emerging technology that contains nanotechnology, material science, and reverse engineering center to work together in convergence. It's a pleasure for me to be here to talk to you, and thank you all, DSI Scientific Network, for inviting me to talk on this platform. And my title is Understanding Human Genetic Diversity for Improved Health. I'm not going to talk on the detail. The content of my presentation will revolve around human genetic diversity. It is distributed, how understanding diversity contributes to improved health, human health, and thirdly, DSI-related capacity building needs, particularly focusing on the need for South-North collaboration. As you all know, human genetic diversity is a genetic variability present within the human species as a whole, and there is a huge amount of genetic diversity that is created entirely by mutation. And the human genome has already been sequenced and available in databases. Uh, Even the first one nearly to be sequenced fully, and a number of thousands and millions of sequences have been added through time to the database one, and the genome is distributed on the 24 chromosomes in the human genome. When we talk about human genetic diversity, it is a custom to talk about origin and the distribution, because understanding of the genetic evolution or the human evolutionary history is very important in interpreting our data. The two major hypotheses are revolving around human origin and the distribution is the out of Africa model of origin and the distribution, where modern human population is assumed to be originated in Africa, and a kind of subpopulation of it has moved out of Africa around 200,000 or 100,000 years before the present. And the second hypothesis, which is less supported by evidence, is the multi-regional origin and the distribution of human population, but the most supported with genetic as well as other lines of evidences is the out of Africa model of origin and the distribution of modern human. I don't know how many of you are in human population genomics, and I hope all of you know about this. And some of the places, including the America, the Australia, are occupied relatively recently in that sense. So the second application of human understanding human genetic diversity is in its application to disease, understanding disease, the backgrounds of disease. There are many different kinds of disease that can be categorized into three in terms of human genetic variation. Mendelian disorders are mainly caused by only by genetic factors, or the interaction of gene with another gene, not too much environmental impact. So these are relatively easy to identify, to sequence, and to intervene relatively if there is intervention at all with gene therapy, and so they are rare in nature. And the second group is polygenic genetic disorders that are caused by several genetic or gene regions, and in addition, a number of environmental factors and the gene interactions contributing to the polygenic or complex disorders, where their understanding is also very complex. And the base human population genetics is very important in interpreting the polygenic disorders. So this includes the common diseases such as the cancer, different kinds of cancer, heart disease, diabetes, and many more are categorized under this particular way. And thirdly, understanding human genetics is also important in understanding susceptibility or resistance to infectious disease. Yes, many of our susceptibility and the resistance to a number of genetic infectious diseases is also governed by genetic variations or genetic sequences. So many of which are understood very well, such as malaria, HIV, including many other viral diseases, are also and our response to drugs in terms of pharmacogenetics is also mainly controlled by our genetic background. So, understanding human genetic variation is really very important in understanding these things. For many of them, um, for some of them at least, there is genomic medicine that is underway, at least in developed countries, and the issue of using genetics or genomics in medicine in low and middle income countries is very rare, and but there are some examples. But in some countries like Ethiopia, even no genetic courses or molecular biology courses are given for medical students, and there is a high gap in understanding the genetic basis or the molecular basis of diseases and so many more. Um, uh, the research parts in human genetics where I have personally contributed as a member of different groups is in Ethiopian context, Ethiopian population genetic diversity, which has collected thousands of samples from across 80 plus language spoken in Ethiopia, ethnic groups in Ethiopia, and this was aimed at understanding the level of genetic diversity and its a relationship with some kinds of pharmacogenomic responses and human history in Ethiopia. And the complex genetic history of the Ethiopian population was understood with a kind of admixture level of relatively 40 to 60% with Africa and the non-African populations so far in general, with different regions. Gene, culture, or language associations have been identified with our recent paper in Nature Communication. And the second line of project where I was a PhD student there is a Lactis project. We collected over 1,600 individuals, sequenced a region of the Lactis gene, and covered many of large populations in Ethiopia and compared with other populations in the global world, and discovered before this study, the notion was that only a single variant causes lactase persistence or lactose intolerance. But after our finding, we found multiple causal variances and so on. The third one is African Genome Variation Project, which was, in that case, our contribution was 125 individuals fully sequenced from 5 Ethiopian populations. All of them are done together with other organizations in the world. And we used DSI in the projects. We contributed DSI from all these projects, and this we generated a database which is stored in open access. People are using our own as well as we are using others DSIs. So I'm going to talk a little bit. I'm sorry if I'm running out of time. Uh, but on this slide, I want to deal with it. Capacity building gaps in the South in general, with focusing in the Ethiopian case. We are, as a biotechnology institute now, we have under the biotechnology center of around 6 departments: the agricultural biotechnology, environmental biotechnology, industrial, health biotech, genomics, and the bioinformatics. Where I am based is in the genomics and the bioinformatics sector right now. We have a huge shortage in the trained human power, particularly in the bioinformatics and the omics sectors. So we would like to get some network and connections to build their capacities in that sector. The second one is in infrastructure. We do not have a single functional sequencing technology currently for 120 million population in Ethiopia. Sorry for that. There is a financial constraint in our local currency. The government assigns a budget, but due to the hard currency, lack of hard currency, we cannot buy instruments and consumables. We have that challenge and shortage in storage and the computing facilities. We have a small server at our institute that is the first one in the country, but currently we are storing some DSI information on this server, but this is not a full capacity server. We want to work with the other group, and we want technical and expertise support in translational research using DSI materials. Particularly, as an institute, we are supposed to give biotechnology techniques to produce products, but we are not really actually doing the modern biotechnology in actual sense. We are only dealing with the traditional ones, but we need collaboration on those areas. And my personal opinion, we need to have collaboration as a south north areas, because we know that the south has got huge biodiversity and the north has got technological and the knowledge development. But don't misquote me. The huge biodiversity is also there in the north, and there are some technical and the knowledge coming also from the south, but collaboration is really very important. Let's collaborate and let's work together to make the world a better place for all of us to live in. Thank you so much.
Thank you, Tamiru, for this great presentation. Then, yesterday we were talking, remember, with the need for infrastructure in the global south, and also how it's really important even for consumables to be available for us to do good science, because we may have good biodiversity. In order for us to understand our biodiversity and to manage it sustainably, we need to have equipment, we need to have infrastructure, but even though we have the knowledge, but we need to have the necessary infrastructure to be able to operate. And also we need really to have good collaboration between North and South, and also between South and South. So I think it's critical. And thank you again for this great presentation. So we will finish this series presentation with Suel, who's going to present lesson learned from ABS capacity building. Over to you, Suel.
And no, it's okay. I just am getting a WhatsApp. Suhel, you're not on camera, so could you switch perhaps with either Anne or Melania? And actually, Tamira was just on the edge, but you're good. So this will be, come here. Just come sit next to me. And then you're definitely on camera. And same thing for the panelists. The view is basically from Charlie to Tamiro. So then we'll switch the three of you afterwards.
We're not done with logistics yet.
Exactly.
All right, let's give it a try. Well, good morning. Yes, Suhail Al-Janabi with the ABS initiative. As you see from the bottom, it's a multi-donor initiative which is implemented by German Development Corporation, by GIZ, and hosted by our Federal Ministry of Economic Cooperation and Development. And we are, I would say, on the spot already for quite some time, doing capacity building, capacity development on access and benefit sharing. And as you see, time goes by, and some phases are still there and negotiating and changed also their roles. I want to quote in that regard maybe Pierre Duplessis, who is, as you know, one of the key negotiators, but he's also quite active in capacity development. And he said, capacity development is like washing dishes. It will never end. And this is probably a very true thing. And what I would like to share with you today are key lessons learned from kind of 15 years capacity development on access and benefit sharing, but that of course, mainly towards a different target group, which are then regulators, governments, to some extent, of course, also the science community and business, but first and foremost, the regulators, and those are also those, of course, that will decide on the future framework on DSI. All right. Just a snapshot where we are mainly operating. You do see that in Africa, that's the key region, but also through other bilateral projects, we supported then activities in other parts of the world, also at regional level. Of course, as you go, you do have to adapt your capacity development strategies. So you see the upper timeline there. We started off in 2005, and we're now in the 2020s. And here, this is more or less the period where the African negotiators in the main at core got technical support in order to be able to negotiate the Nagoya Protocol. And you do see on the right-hand side the different means of implementation. So with peer exchanges, with issue-based trainings, coaches, and so on. So, I mean, we also need to consider this in DSI capacity development, what the modes of delivery are going to be. And of course, there's a variety of modes of delivery. So that was very much from the sub-regional level into the global level, but also at the national level, to support the negotiators in understanding the core issues before the Nagoya Protocol at all was negotiated. And then, of course, as we went, there was a focus on ratification, as you see. For that, you needed to dive deeper into the national level, but first results were also to be attained in terms of benefits, value chain development, and so on. And now we are basically in the implementation period where we focused on three core processes, if you so wish. It's to support at national level the development of institutional regulatory frameworks. So the cornerstones to operate in. It is also to support indigenous peoples and local communities. And then the development of ABS agreements, non-commercial and commercial ones, and as much as possible also to reflect then the IPLC's roles in that. So that's a bit where we're coming from, and I think just to run to some of the key issues that might be also of relevance for DSI capacity development in the future. We are dealing with countries that have very, very different setups in terms of their ABS regulations, their frameworks, and some are still developing their frameworks, others are revising it for the second or the third time. There is maybe a good understanding now of the legal aspects at the level of the focal points, but not necessarily at the other line ministries, for example, here in that regard, also the ministries of research and so on. And whereas there is understanding, yes, on let's say the core issues that are related to the legal aspects, questions in terms of monitoring the utilization, to understand the compliance mechanism of the Nagoya Protocol, the interaction with the CNAs in other countries. This is quite often still as an infant stage, and what surely is also of importance for the future DSI debate is that we still have to work with an inadequate IT infrastructure. We do see that focal points and others, if they want to take part in Zoom meetings, that they're still going to internet cafes because landlines are not in place as they should be. They're doing it rather than on their mobile phones. And I think this all needs to be taken into account, that we are entering a different world here in that regard. If we looked at IPLCs, and we heard that as well, also here the rights of IPLCs in countries can differ from basically no acknowledgement of indigenous peoples to the level that they have a full say, that they are basically that pick and mud to be obtained and negotiated with them. We do see, of course, that still all the concepts of valorization is unclear. But on the other hand, we see it at the level of more the commercial, but also the non-commercial side, that there's a tendency to avoid access to genetic resources where there is TK linked to it, simply because of the complexity of obtaining prior informed consent. And that might be also something which is important when we look at gene sequencing in the future. So there, this dialogue and the full integration of empowerment of IPLCs in the ABS arena is as much important as, I think, also now for future DSI capacity development. Specific regulations and procedures. I mean, whereas we do have laws and regulations, implementing regulations and also flowchart process descriptions are very, very rare. In Africa, we find on the clearinghouse maybe some 8 or so, and that doesn't make it easy for a researcher, be it a national one or an international one, to find the way through this permitting jungle. And on top of it, you would need to consider that you don't only need to deal with one permit. I mean, you have to have a research permit. There might be an export permit if you want to get also your genetic resources sample outside for further testing and so on, material transfer agreements and so on. And you would also need to know, of course, in in what sequence, nice word, you would need to have that. So is the research permit a precursor for the export permit and the other way around? That's not that. It's rare that this is already defined on the clearinghouse, and that you can get a good indication in that regard. When it comes then to MUD, the core of ABS, if you so wish, it is the case that mainly your counterparts do not have access to contract lawyers, but basically public lawyers are doing the job, which come from a totally different background, don't have an idea on value-adding steps, on industrial IP, on research IP, and so on, which may also be an issue for DSI in the future. And with that, it comes also this aspect that there is a little indication, because key elements of mutual agreed terms, in particular, of course, the benefit sharing clauses are confidential. So questions of expectation management, is it 1%, 3%, 5%? There are no real orientation figures, yeah? And that's probably also in terms of capacity building on DSI, scientific collaboration, what is it that one could expect from the partner? There is not a lot of disclosure on that, and that also impacts now here on the negotiations target 13 indicators. How do we measure at the end of the day impact of access and benefit sharing, which is, if DSI would fall under that, is also invertibly linked to that. There is the monitoring of ABS contracts, which is, of course, something that one needs to take into consideration, because there's a point of access, and then value addition, research and development. Maybe a product is only in pharmaceuticals, also in biotechnology, some 15, 20 years later on the market. So focal points do have to store of course, all these permits that they have issued, and there is, of course, a turnover in terms of personnel and so on. And it's going to be a challenge that for permits that are going to be issued today, that they are still on the hard drives in some 15 or 20 years, when basically the checkpoints would then, be it in scientific or in commercial applications, give a feedback to those countries, and then provider countries would have the possibility to compare. So maybe that's also something where, in terms of hardware and so on, one could work together. ABS compliant value chain development, it's still a myth. As much it is for you guys, the scientific community or the commercial community, a myth and a secret, how the different ABS regulations in countries function. I mean, I told you that the difference in scope, the difference temporal scope, physical scope, in terms of also questions of procedures, as much it is a mystery to focal points, how the different sectors function, and also how the different scientific landscapes actually do publish, in which country, what kind of obligations a scientist had. So there is also still a lack of that understanding, and there is only, and we heard that from Melania, not in many countries there are national valorization strategies, be it on biotech, be it on biodiversity economy. So also here, of course, this element of using DSI towards a biotechnology oriented society, we do find that, yes, for example, in the 2063 vision of the African Union, but it hasn't yet trickled down at national level. So that's also something that one would need to take into consideration. Well, last but not least, of course, that's what we are dealing with now for maybe some 3 years. It's of course, capacity development on DSI, mainly vis-a-vis, of course, the regulators. And there are still conceptual misunderstandings, and you can see it up to now in the negotiations. What is it that we are talking about? Is it the sequencing from a genetic resource to DSI, or is it about the access of DSI in the public databases? And this is still mixed up now when we're talking about hybrid systems and multilateral system. Would it impact on national sovereignty? So that's definitely something where more dialogue is needed. And this is one, of course, of the points that I also mentioned, that there are barely interministerial dialogues on the one hand between the ministries of research and the ministries that are dealing and regulating ABS. On the other hand, also when talking to CNAs, they're not really aware of their DSI constituency back home. Yeah, so who are the universities? Who is dealing with that? I don't know to what extent, for example, our colleague in Ethiopia is, for example, in exchange with the regulators there. So we also see that there's national processes now to regulate DSI for the good or for the bad in terms of open access, but at least you would have clarity on how things are dealt with, are basically stalled because we have the international regulation negotiations now ongoing, and that's also something that we would need to consider. What is it that we're going to do now in the meantime until we found a solution in COP 16 or 17? Yeah, that's there is to be some smart thinking necessary on how to bridge that very gap. Yeah, so this is a snapshot of some key issues that I think are relevant also for future DSI capacity building, and that's just a little bit of an outlook that now, of course, the initiative will take into account the elements of the global biodiversity framework, questions of monitoring, much more going into multi-stakeholder partnerships, maybe also some on DSI for showing proof of principle that collaboration is feasible and that there are benefits, monetary, non-monetary, to be shared, and that also a lot of mainstreaming is necessary. That could also be mainstreaming scientific agendas into the ABS and environmental agendas and the other way around. And of course, that takes also up questions of TK, embodied TK, all these discussions when we also have one component looking stronger at the questions of indigenous people and local communities. Thanks a lot, and I don't know whether this was off topic, but it might be useful then for consideration along the way we will go in terms of capacity development, because you can't do one without the other. Scientific capacity development, technical training, and so on needs to go alongside with also then better understanding of regulators of the issue, so that we're getting more from a regulatory to a facilitation mode. Thanks.
Thank you. So we will go. We will have a just 5 minutes session for questions for the four speakers. So, okay. Questions. Question. Okay. So we will go to the panelists now for the session. Thank you so much.
So what I'd like my panelists to do, I'm going to take Charlie's seat since I'm moderating, and then I want the four of you to be in my chair, and then the three next to me. Sorry, yeah, we're kicking you out, Sohel. Move. You should go back to your corner. Awesome. Thank you. Sorry, Charlie. And then we can kind of do maybe a little semicircle-ish. I mean, we'll see. So for those of you in the room, we're trying to accommodate the camera in front of us that can only see a very narrow bandwidth because the camera doesn't move or spin. All right. No, Charlie, you too. You come back over here. Oh, yeah. And Tamiro, you stand up. Your chairs are live. Yeah. No, you sit here. Exactly. Sorry. Little musical chairs. This kind of freshens it up, though, doesn't it? Right? All right, so I'm going to introduce the panelists while they are shuffling around. If I can find my notes. So here, who's sitting next to me, right next to me? I'll try and adjust here in real time. So I have Dr. Guilherme Oliveira. I'm going to save the doctor titles because they all have doctor titles. So Guilherme Oliveira graduated in biological sciences from Universidade Federal de Minas Gerais. I assume that's in Brazil. Yes. He obtained his PhD from the Department of Biology at Texas A&M University in the United States, and then was a postdoc there as well, and at the University of York in the United Kingdom, and at Fiocruz in Minas. He is currently the scientific director at the Vale Technology Institute. Then Charlie Mabala, he self-introduced, but he did his PhD in Nebraska, biotech at Nottingham, and then also at INRA, and now in Gabon, and where he's part of the delegation here today and works with the Nature Conservancy. And then following him is actually my boss, Jörg Overmann, who's the scientific director at the Leibniz Institute DSMZ. He is a microbiologist by training, did his PhD in Konstanz, a professor in Munich, and been at the DSMZ since 2010, where he leads a real big ship from the world's most diverse microbiological collection. And then finally, our colleague from France, Dr. Martine Hossertz, did her PhD in science and worked at the Center for Functional and Evolutionary Ecology, which is CNRS in France. And from 2018, she has been working at the Institute of Ecology and Environment in Paris. So all of these people bring different perspectives to our panel, and you heard some incredible talks today. So Melania started us giving an overview of the different ways that DSI capacity building ties into the bioeconomy, to sustainable development, to conservation. Then from, I'm losing track, from Anne on the intersection of biodiversity genomics, IPLC data management. Then from Tamiru on genetics and health from humans, and then from Suhail on lessons learned on capacity building in general. And I would like these distinguished panelists to kind of follow up on that. So Guilherme, I'm going to turn it over to you first. And I would love if you could give me a sense, you saw the programs that Melania introduced, and we kind of see the spectrum from, bioinformatics training courses, and then we see sort of, whole labs and infrastructure being built up, and kind of a span of different types of capacity building. What in your experience works the best? What have you seen in your experience in Brazil, working with colleagues in Latin America, about what's like low-hanging fruit and what's high-hanging fruit that requires more time, more investment, and what's your opinion on what we need first and then next?
Okay, so as reported by our colleague from Ethiopia, the landscape is quite variable from these places like he described to places like in Brazil, where we do have training in bioinformatics. We have excellent bioinformaticians at hand, but the infrastructure is pretty much spread throughout the country, and that's very inefficient. So in order to get there, I think we need to make sure that we are able to carry the work from collecting samples to sequence these samples, analyze the samples, and then the next steps to make them useful to humanity for science purposes and also for protection of nature and bioeconomy development. So the key there for me in some of these countries is how we build this infrastructure where it can exist, but it still doesn't exist as a robust, strong, well-financed and long-term financed infrastructure. And that's what we are trying to build now in Brazil. Other countries may need to get together to do that, as we heard from our speaker earlier. Well, it is a substantial amount of money, but it's doable. And I think governments also need to realize that, to my taste, this is almost the case of national sovereignty, because I mean, if the biodiversity is there, we should be able to understand it and use it to our best interest, and also for sure many of these species will have relevance globally. So I think I'll start there with we need to get together and make sure that building this infrastructure is a need, and we need to move fast, because as we all know, species globally are disappearing faster than we can understand them. So the books are being lost.
Yeah, we saw that from Anne's presentation as well. Did anybody want to follow up on that, or should I? Yeah. Mm-hmm.
Yeah, the thing is, Brazil is a very big country, so I can understand that you want to build one big infrastructure. But for example, I think in place like, I would like your advice on that. But for example, place like Africa, I think regional infrastructure will be better than one country, one infrastructure, because of the lack of manpower, as you said, and also the lack of training people there. So I think sometime in some situation we need to think about more regional than country type infrastructure.
Charlie, are you adjusting the mask, or are you following up on that? I mean, you also work from Gabon. Your experience there, the
Yes,
regional versus the
I think it's critical to have like the sub-regional dimension. I think it's, if we want to be able to to work and have sustainable results. I think it's very interesting for countries like we want to, for example, to manage the Congo Basin. It will be very important for the 10 countries that are part of this Congo Basin to have infrastructure that will be allow them to be able to perform and to understand the biodiversity and to manage them sustainably. So I think it's critical that we also have that sub-region, some kind of approach in order to achieve our goals.
Yeah, that's fascinating. I'm writing down 10 countries, exclamation point here, because so the Congo Basin is not the Democratic Republic of Congo, right? It's not that simple. It's 10 countries that, if you're interested in the ecosystem, then the geographical boundaries don't matter. I mean, they matter for the politics, they matter for the regulatory infrastructure, but if you're asking the scientific question, you need to involve 10 questions if it came to building up a Congo Basin thing. Would that be true though, Guillermo, coming back to I'm thinking about the Amazon, also more than Brazil, right?
Absolutely more. 40% of the Amazon is outside of Brazil. So I think 40%. Yeah, I think we have a good case for South-South collaboration as well, because we understand our problems perhaps a little better, and we can join forces to solve them. Now the keys were to find the resources for this. We had Brazil reporting that a lot of funds have been promised by the rich nations to support conservation of biodiversity. And I think conservation of biodiversity, I think some people would agree, goes beyond protecting the native species, but also we need to realize that people live in these areas, how they can best benefit from it, not only by the resources they can extract, but by transforming and revealing the genetic diversity and using the technologies that we all know are currently available to bring it to fruition.
Yeah, we have, the buzzword here at the COP and CBD world in general, or actually maybe environmental policy in general, nature-based solutions, and that kind of goes to that. And Jorg, I want to come back to you. So this first part of the panel, we're thinking about DSI, capacity building for biodiversity, for conservation, Congo Basin, Amazon Basin. But, you and I at the Institute, when we talk about bioeconomy, we as microbiologists are always saying, it's not just corn, it's not just bioethanol, So tell us what you think about the need, the possibility for the use of DSI to, lead to better paying jobs, to build up a bioeconomy, to be sort of an economic engine. What are your experiences and thoughts, both when you've worked in Latin America and Africa and what you're seeing in Northern Europe?
Yeah, thanks for the question. So maybe I start with some practical considerations which I came across, and I, for instance, can fully agree to what Tamira has said about Ethiopia, where we have been working for quite some years now, where it is really the basic bioinformatics, the access to data, servers, if they're there, they can go down because of the power supply, these type of things. And we have all this range to what Guillermo has shown. I'm working a lot in Colombia. There's a completely different situation. So what we need probably if you consider bilateral work, we need to consider the specific needs. And one way to do that is focus on specific projects which address, for instance, also the needs of small farmers. Like we are now thinking about coffee fermentation project with small holder farmers in Colombia, which would give them the opportunity to produce more high quality coffee more reliably, and so secure their incomes. These types of approaches, I think, would be helpful. But that would mean on the other side, and now I'm speaking as somebody on the side of the European funding, we would have to have tailor-made or well-tailored funding schemes, which would be different whether we work, for instance, in Ethiopia versus where we work with Colombia. So that would be an important need. Now we have categories which may not fit that, exactly that demand. So that would be one. Also, so besides of the bioinformatics and so on, I think it is clear that sequencing now is not the bottleneck, really, technologically speaking. We need we need facilities. I would like to point out that one global sequencing center probably wouldn't really do the job. We have experience in Germany. We have a network of sequencing centers which were built by the German Research Foundation, and that works quite well. So I think a well-established network would be an opportunity to do so, and then you can outsource. For those countries who can't run that within their own borders, you can outsource. And mirroring the INSDC, we have heard about that. I think that would be a very good start to provide the access. But data literacy and bioinformatics, I think that is the most frequent needs and what we deal with in our bilateral project the most.
Yeah, that's super helpful. And what I mean, I remember this. We've written grants together. It's completely unsexy, for a policy situation like this. But actually, how a grant is advertised or is written by government makes a big difference. So there can be not only there can be the best will between the scientific community, but if the grant doesn't allow, for example, for technology transfer, if it's explicitly excluded in the grant scheme, then you can't bring your PCR machines, your sequencing machines, your servers over or buy them there if the grant excludes that. So these administrative aspects have to be considered when we think about international collaborations. And Charlie, we were talking yesterday too, you brought up other sort of administrative or practical challenges that you've seen in Gabon, ranging from, give us a sense of other kinds of practical things that maybe are a little bit less technical, but still equally important.
Yeah, for example,
Microphone.
Oh, sorry about that. Yeah, for example, if you want to apply for a grant, the technical, like the language barrier, also it could be be a key. But I think that also the fact that we are really looking for not only to have capacity building, but also have the technology, the transfer of technology, which I think is critical, because we just just don't want to have just PhD student being trained, of having some government staff going to get some training, but we also have to need the ability to have the technology to be present so that we can manage sustainably our biodiversity. I think it's critical, and Virginia was saying this in this presentation about the fact that even for consumables, that's critical, and then we don't have them to do the work. You could have a machine, but if you don't have consumables, how can we do the job? How can you actually understand and be able to publish on time and get the scientific recognition for the work that you're doing? I think it's critical, and it's very basics. It seems to be very basics, but I think it's critical for us to have it. So.
Yeah. Exactly. So for those of you that aren't from the lab world, when we talk about consumables, we mean like the plastics, the chemicals, the tubes and liquids that we need to be able to do molecular biology work. And what's been interesting within the network, we've had these conversations in a working group, that it's actually more expensive to like twice the price to buy these consumable lab reagents in many low and middle income countries than it is in Northern Europe or the United States, right? So it's like, how could that be? Because the price of everything else is generally lower there, but for these sort of high value products, because of import tariffs, et cetera, it's actually much more expensive. So it's like the opposite of what you would expect, even though the cost of labor might be lower. And these are big barriers to sort of budgeting into a project application, et cetera. Martine, we weren't able to have a colleague here from the Asia Pacific region, but you've done a lot of work in China. Can you give us a sense, are we representing some of the same issues here, or is it a bit different in the experiences you've had over in China?
In China, it's a little different, of course, because the main experience I have is about the work we have done with the Chinese Academy of Science, because the CNRS has an association with the Chinese Academy of Science, and we were doing some capacity building with in Sichuanbana with a Chinese tropical garden there. And because we are specialized in the genomics of floral volatiles emission, so they want to be able to do that also in the garden. So one student came to our labs, and he was able to do everything, to learn everything, and he has a very good background in chemistry and a very good background in bioinformatics. So it was totally different situation. So when he will be back in the garden, he will have all the equipment because he can write grant, because they know how to do it and everything. So with China, they need some capacity, but more like and training and intellectual capacities than, for example, I work also in Senegal, and the situation is totally different there. So
Yeah,
because we have also
shades.
students who came to our lab, but after that, when they go back to Senegal,
Yeah.
nothing that they can do there.
Right,
So,
to be able to create also opportunities in country for the return, to prevent the brain drain, right? So that's taking account to sort of the different levels of development. I'm keeping an eye on time. We had a logistics issue here in the room. What I'd like to invite the audience is to think about, if you have a last question or two. I'm going to ask for a last word from the panelists. And then I'm also going to kind of look around the room and see if there's any questions that pop up, if you catch my eye, while they start to take the floor. So panelists, I would love to have you kind of think about, after this discussion that we've had, what needs to happen next, in your opinion. So we have the parties are negotiating in DSI. 6 options formally in the annexes that you can find them. And of course, option 4 is the most popular option that has zero conflict or disagreement around it. But yet, unfortunately, we have no details in any of the negotiated texts on what option 4, capacity building for DSI, might be. So what do you think these negotiators representing us here at the meeting should be writing in the text over the next few days? What do you think is important? I'm going to, should I go to you, or where do you want to start and go across here? You want to go back to Martine? Yeah?
Yeah, I think what I have seen, training is really a big thing about capacity building, and it's not only training about bioinformatics, to know about the omics. It's also, as you said, it's also the way to write a grant. People, I think from Senegal, even the main professor in Senegal doesn't know how to write a grant for EU. It's so difficult, and it's culturally totally different. The value, for the European country are totally different than the value in Senegal. So it's also a lack in training for the students there. So, and we can teach them how to do it, or at least how you want to do it. So training, it's a good point that we can build on capacity building.
Yeah, so you go back to basics then,
Yes.
really back to basics.
Yeah.
So it's not even, the training, yes, but training even in the most sort of basic scientific skills, administration and stuff, right? Jörg, add on to that for us, please.
Yeah, of course, I fully agree. As I said, I just want to point out maybe 2 things. If pharmaceutical products are a big share of bioeconomy, and if 60% of the patents affiliated with organisms are from microorganisms, and we have to consider the needs and the ways to exchange this knowledge and to deal with the data, and DSI is a big issue here. So I think there's no way around a multilateral solution there, I'm pretty sure, because of that simple fact of a global distribution of microorganisms. But I want to make a second point. Most people talk about sequences. I think we will rapidly see in a completely expanded set of data, and I'm talking far beyond what we have heard about the metadata in Anne's talk. I'm talking about phenotypic data, things which you can exploit much more rapidly than going back to the sequence, analyze them yourself, inference of phenotypic data from genotypic data, and so on. So we're going to see this huge expansion, and we have to make sure that is from the start being accessible to all countries, especially those who want to develop the bioeconomy. So not only multilateral and open, I think we have to make sure that gets developed right. And then bioeconomy will be much faster to develop than we have seen it before.
Can I ask you to just jump in and give our listeners a little bit more concrete on what you mean there when you say phenotypic data? Are there like buzzwords that we should be listening for? I mean, I'm thinking of do you mean metabolomics, or do you mean even broader than that? Do you mean structures, or what do you mean when you say phenotypic data? Phenotype, by the way, is the expression, the functional expression of genes. But I want him to give us more lay people buzzwords here.
So if you were able to ask or to find any organism producing some sort of compound or doing a process, yeah, for instance, bioremediation of a contaminated soil, say, for instance, you could go back to the genome and do your thing, and 5 years later you might have the proper source and have heterologously expressed the gene clusters and blah blah blah. Or if we fill the databases as quickly as we try to do that now, with that information, what is the type of bacterium, for instance, or fungus performing that reaction? Where do I find it? What are its properties? How can I grow them? Can I grow it to big scale and inject it in soil? That type of thing. This type of information will speed up, will allow leapfrog developments in all of the countries, because you don't have to go through the biological analysis first. You can exploit the knowledge which is there and combine that. So KI, all these things, artificial intelligence, all these tools, it's about tools, it's about pipelines, it's about computer codes. It's less so than about sequencing. And I think that's where part of the future will be in the next years.
Yeah, like bringing together multiple types of data sources into multiple different abilities to question, to mine the data. So I
Right.
sort of hear you saying it's not like any one magic bullet kind of data,
No.
but it's rather the synthesis of many data types into the ability to ask functional questions. I think that's really helpful. And we've talked a bit in the DSI discussions also about the importance of interoperability of data. And we're not saying that because we're geeks and we want to protect our databases, but we're saying that's where new knowledge is going to arise from. Charlie, your last thoughts. What are you leaving here with?
I mean, I would say something related to what Sue said about the administrative burden. I think in capacity building, that's something that will be taken into account. And I think that also this whole government approach that will be very important at the national level. But also, I would mention that Africa has a 60% of population with young people under 25.
Wow.
So I think this is a massive
Labor force.
labor force
Yeah.
for DSI, and then you do really involving database that are really growing really fast. So we have here the opportunity to train our young people to be able to master this whole knowledge and to play a key role in what we want to do with with DSI in the future. So I think that it's really critical that well beyond just the capacity building and the technical aspect, also the administrative aspect also to be taken into account seriously, and also very inclusive of youth and also of the IPLCs. Thank you.
Yeah. Yeah, you hit a lot of buzzwords there. Okay, but I love it. No, I really do. So administrative and technical aspects. And we have a really strong youth delegation here too. So maybe we also need to get them to mobilize also internationally and sort of think about the opportunities of the future. Guilherme, what are you leaving here with? Is this resonating with you?
Absolutely, it is. And the two bits that I wanted to add here is that I would like very much to see in these documents a free environment to do research. I think regulating access to DSI to do research is not just, I have a hard time wrapping my head around how this can be done. So why do that if you cannot manage to police it? The other bit is that talking about DSI, and in a broad sense, I like very much what my colleague mentioned. It's just not sequences, but in a broad sense, our countries in the south, where most of the biodiversity is, we need to get a handle on how we are going to produce the DSI. Because if we can't produce it and ourselves, I mean, it'll be hard to share benefit if it doesn't exist. So we need to get that going. Thank you.
Those are two great take-home points. I was looking around the room throughout the panel, seeing if there were any questions. If anybody's brave. I know this has been a very different morning with the Friends of the Chair group on DSI meetings, so we don't really have a diversity of participants as we might have originally hoped for, but hopefully, I think this was actually streamed on YouTube, which should mean that it's available even later, so people can come back to this. And also, if you would like to receive any of the presentations from today, we are happy, I think, most or all of us to share presentations and give you any more details on the things we've talked about here today. with that, I would like to close up I'd like to transition from the panel, hand it back over to my co-host Charlie, and to wrap it up for you guys. Go ahead, Charlie.
Thank you, Amber. So I would like to thank you, all of you. I'd like to thank the organizer and also the presenters for interesting presentation, all those good insights. So we hope that at the end of this COP15, we will have good results in terms of DSI and how we manage DSI, how we access DSI, and how DSI is being implemented globally, but also at the national levels for all the different countries and the parties. Thank you so much.
Thank you.
Bye-bye.
Good morning, everyone. Hello. We are going to start. We welcome you if you want to get in the table. There's a lot of space here around, and maybe it's more, it's a better idea to see each other. So if you want to move from the back, you are welcome to come to the table so we can start. Thank you very much.
Oh, okay, yeah. Okay, good morning, or welcome to the session 8, Mobilizing Tools and Technologies to Support Implementation and Monitoring of the Post-2020 Global Biodiversity Framework towards a Global Biodiversity Observation System. My name is Britta Garfield. I'm with the Smithsonian Institution. This session is co-hosted by GeoBon, the Humboldt Institute, and the Smithsonian Institution on behalf of the Consortium of Scientific Partners. And as you all know, of course, the monitoring framework is central to the Global Biodiversity Framework. Its indicators are vital to track and report on progress towards the goals and targets, and can also inform scenarios that forecast and guide the planning of policies and actions. We will have 5 keynote speakers who will provide an overview of existing monitoring tools and the status of the monitoring framework. Then we will have a Q&A and then a panel discussion. And we invite you all to ask questions and participate in the discussion as well. We will address key aspects of the science needed for the implementation of the monitoring framework. First, filling the thematic and data gaps. Second, establishing a detection attribution system so that we have indicators capable of responding to the actions implemented to reach the targets of the post-GBF, post-2020 GBF. Third, strengthening the capacity of countries to implement biodiversity monitoring systems and generate the data needed for the indicators. And fourth, networking national efforts to achieve a global observing system, supporting continuous assessments of progress. So I would like to hand it over to the first keynote speaker, Professor Alexandre Antonelli, the Director of Science at Kew Royal Botanic Gardens. Professor Antonelli studies the distribution and evolution of species and develops methods to speed up scientific discovery. He focuses on drivers of biodiversity change through space and time, among other topics, and is also in interactions with society and scientists across disciplines with the main goal of increasing knowledge, awareness, and the protection of biodiversity around the world. Over to you, Alex.
Thank you so much. And good morning to everyone. Nice to see you all around this table.
some people perhaps online, just behind me as well. So very, very nice to see so many people gathered today to talk about methods and how we best can fill the gaps in data and knowledge we have, but also track progress towards delivery of the new global biodiversity framework. I want to start today by showing you this beautiful image from the Atlantic forest in Brazil. It's a place where I visited many times as a kid, growing up in Brazil and then going down to the coast, passing through those amazing forests. But unfortunately, this is a picture I took a few months ago from the same place. And last year, deforestation rates increased by 125% in the region. And now we have less than 8% left of the Atlantic forest, which once covered the whole of eastern Brazil. So that's the major challenge faced by biodiversity on land today. So the change in land use and deforestation, another change to natural habitats. But of course, we also have the challenge of climate change. And in each one of those stripes here, you see how temperatures around the world have been increasing. And that's, of course, something that puts species at risk, even if we protect some of those fragments of forest and other habitats as well. So my talk today is about how we could use new technologies, and in particular, artificial intelligence, machine learning, and also data from remote sensing technologies and citizen science to help us understand and protect biodiversity. And my apologies for some of you who were here in the same workshop yesterday. I just want to repeat a couple of things, but go a bit deeper into the methodology, which I think is the core purpose of today's workshop. And I want to give you a couple of examples of the technologies we've been developing in order to pursue that goal. So the first one is a method which we're calling conservation area prioritization through artificial intelligence. And what we've developed is a system where in each one of those cells, and you can think of the system as a country like Madagascar, or a region or a county, where in each one of those small cells, there is a number of species, a number of individuals of that species. It could be plants, but it could also be marine habitats, for instance. And if you protect the area, which means you reduce the disturbance, which is in form of degradation or building roads or deforestation, you protect the species and individuals within that cell, which is the dark black circle or a square around a unit. And what we've been doing is to develop both simulation approaches and empirical validations to identify what is the best combination of areas to be protected or managed for sustainable use to protect as many species as possible. So basically focusing on the outcome. And this is a technology actually borrowed from gaming. So if people play video games, that's something that they use a lot. So reinforcement learning. And it's also something used a lot in the financial systems where you basically learn by doing. So it's not a predictive model to foresee what's going to be the likely outcome, but actually you make the decisions as you gather data a long time. And that really outperforms any kind of existing technologies for systematic conservation planning and for deciding where to protect, and really takes into account the complementarity and connectivity among those areas that are managed. And if you do this, you can either focus on the biodiversity outcome. So for instance, how many species you want to have at the end of a period. So by 2030 or 2050, how many species we will likely get. But you could also focus on something else, like the amount of protected area. And in this case, if you do that, you significantly decrease the number of species you're going to save. And that's because if you protect the cheapest land, not the land that is most important for biodiversity, you check the box of protecting as much land as possible, but then you significantly get a worse result than actually even random protection of areas on the system. So it's very important to be able to play with the data and actually look at the outcome of those simulations and also the empirical validations, which we are seeing in several systems now. And that's one way of incorporating the complexity of systems, because it's about climate, it's about the species distribution, it's about migration and many different variables. And some of the variables being discussed now in terms of metrics could be easily incorporated into this kind of framework. Because especially when it comes to neural networks, you can add lots of different variables without adding statistic error to the analysis. So if anyone will be interested in that, I'll be very happy to talk further about this afterwards or during the workshop. My second example is about speeding up red list assessments. And we've seen talks yesterday about the IUCN red list of threatened species. And that's one of the major tools for understanding the threat that species face. And those are full-fledged assessments. And we at Q, we've been doing about 1/3 of all red list assessments for plants. And they're really powerful. They're really important in terms of understanding what are the likely threats that particular species face. But they're also quite resource consuming. It takes at least 1 day for a skilled botanist to actually assess each one of those. And there are tens of thousands of species which haven't been assessed yet. So we've been trying to explore the opportunity of using machine learning to learn from the assessments done by experts and to identify the likely threat of species which have not been assessed yet. It is not a replacement, and I really want to stress this. I think the IUCN assessments are still the golden standard for this, but it's a way of identifying the species that are most likely to be either very threatened or not very threatened, so that we can focus resources on those that are more likely to be at risk. And for instance, in Madagascar, we've just recently done that. Last week, we had 2 papers on the diversity of Madagascar. And we also seen that lots of the new species been discovered to science, and that's what the plots on the right show. So the discovery rates through time, and you see it's nowhere near to be flattened out. So every year we're discovering so many new species to Madagascar, and many of those are actually very likely to be threatened from the beginning. So what we did was to do a comprehensive survey of both plants and animals for Madagascar. And what you see, the black line here in the middle of this diagram shows where the threatened and non-threatened categories are divided. So basically everything that is above the black line are not considered to be threatened, and all the species in those many different taxonomy groups below the black line means they're likely to be threatened. And in some cases, like for ferns, the dark green color, so the first plot there shows the current assessments by IUCN Red List and its partners. And you see here that most species seem actually not to be doing quite well, but when we use the machine learning approach to all the hundreds, so 270 species here which have not been assessed, the overall picture, the outcome of that is that most species, or perhaps half of the species, are actually threatened. So this is a way of speeding up and concentrating our efforts where they're most likely to be making a difference. So now the experts can actually look at those species which are likely to be threatened but not yet assessed. And that's something we could roll out to many different systems and also do that on a frequent basis, because understanding not only the threat to species but the trend in threat, so how it changed over time, is going to be absolutely critical in the implementation of the new global biodiversity framework. My third example is about how do we fill the gaps. And I know many people sitting around this table in this room are very concerned about this, and very many suggestions have been put forward. I just want to mention an example of the Life Plan project, which we've been involved in, setting up new sites. So this is a global initiative comprising more than 100 sites across the world to make inventories using new technologies for as many groups as possible. So here, just on the left, you see the different kinds of technologies we are using, or methods, including insect traps like the Malaise trap, cyclone sampler for capturing both spores and pollen from the surrounding region, soil samples, camera traps for images of anything moving around, like animals or birds, and then using AI to identify, or machine learning to identify the species there, and also audio recording, where we can record both birds but also bats, and identify once again using the machine learning approach. And for both the Malaise trap for the insects and for the cyclone sampler, which is pollen and spores, as I said, we are taking environmental DNA. So we're sequencing everything in there, which means that in all of those, you actually don't need to be an expert. And we know that there's a shortage of taxonomic knowledge in society. So we don't have enough people to make comprehensive inventories of biodiversity, and it's changed through time. And therefore, this kind of new technologies allows us to actually get some really robust data-rich approximations of the overall diversity in those areas and how it changes by just using those technologies. And then, of course, we still need experts to help guide and identify and classify the training data sets for this. But then at the end, you do get a species list for each one of those sites. And it can be really fun and a way of engaging people in harnessing biodiversity data. So I just show you three photos here. The first one showing how environmental DNA sampling can look like. Just take soil samples in this case. There's some good examples also using freshwater and marine water samples. citizen science, so mobile apps, and then remote sensing, where we are now flying with drones and multispectrum lidars, which basically scanning the landscape, identifying single species of trees and other organisms through remote sensing technologies. So I think the discussion here, and we were asked again to bring some topics. I would love to talk about how could we all put together the different technologies, the parameters, metrics into some system really which encompasses all of those at the same time. And I do believe that the machine learning approach here could be a potential candidate for that. So for instance, in this case, the picture on the left shows deforestation, like the first picture I showed you today. And if you know that a species was distributed across the square there, but now there is a sudden deforestation affecting some of them, you could immediately go from one species not being threatened or being endangered to being critically endangered by just the sheer information gathered from remote sensing. So that's the early warning system that I mentioned yesterday as well. And I think that's exactly what the kind of thing we need, so that we don't wait too long and 10 years until we see that many of those species actually have gone extinct. So I think looking forward, I do believe that AI has a huge potential to help monitor and guide conservation in a dynamic world. I think we've demonstrated that to a certain extent, but there's much more validation and improvements that need to be done. My colleagues and I are very keen to help. So we don't want to reinvent the wheel. We don't want to compete or replicate efforts, but I think it's really an opportunity here in this symposium and beyond to be talking about collaborations and how we can join forces on this. And so how could we create synergies? And I hope that we can have a discussion about this later today. Thank you.
Thank you so much, Professor Antonelli. So next, and we will have an opportunity to ask questions after the presentations. So next, I would like to ask my colleague, Dr. Stephen Canty from the Smithsonian National Museum, actually from the Smithsonian Environmental Research Center. I would like to ask him to give a presentation. So Steve coordinates the Marine Conservation Program at the Environmental Research Center. And he's also the Seascapes Science Advisor at the Working Land and Seascapes Initiative at the Smithsonian. And his research supports decision-making of the management and conservation of marine resources, especially on mangroves and small-scale fisheries. He's been working in Latin America and the Caribbean region with communities and governments to co-develop solutions that provide benefits to both people and nature. So the floor is yours, Steve.
Thanks very much, Britta. Hi, everybody. So I've got a slightly different angle on some of this. I should be presenting more about my work, but having listened in for the last week or so and been talking with people, I've been feeling that there's a very high level of what we've been doing when we've been talking about how we're going to do the capacity building around getting the post-2020 framework in place, and that actually a lot of the things that we're going to be working on are going to be in at the local level, so local marine stewardship. And just here, there's sort of 11, 12 different ways that local communities in marine systems are going to be wanting to manage or protect their resources. And how do we find ways to get their aims and wants of how they want to conserve, protect their biodiversity, and how does that then align with what we're doing at this level at the global framework? And so just thinking more about that, and then thinking, how do communities link in with these essential biodiversity variables? What does genetic composition mean to a fisherman in Honduras? What does it mean to someone replanting their mangroves in Suriname or Southeast Asia somewhere? And so realistically, what are we going to be doing to move that from this higher level down to these communities and then back up again? Because it needs to be this data flow up and down, because what we're seeing is with what we have with these indicators from GeoBond, some have been endorsed. Hopefully there'll be a whole bunch more endorsed and into the framework now. But it always seems to be this upward flow. and all of these indices work in, but how do we bring that down so that the indicators actually mean something to the people on the ground that are doing this, and it's not people like me with the science, it's people who are relying on these resources for their livelihoods and well-being. And so this is something that, community-based monitoring and citizen science, there's a lot of examples where they do fit into these EBV classes and that they work at different scales. So, the evidence is there that we can do this. How do we now promote that out? And so, I mean, a lot of us know all of these barriers to implementation of these observer methods. And there's a lot of monitoring efforts out there where, they've sort of died a death because they're not given resources over the years. People don't know how their data is being used, and it's often in open access and being used by other people for different things, but not to give them the information. So how do we bring that back down? And then the geographic bias, I think we're all aware of this, but also just to remember that even the data that is collected in some of these countries where there is, in the more tropical areas, say, where there's more biodiversity, the data is generally housed outside of that too. And I also think that there's these issues, say, for Colombia and Ecuador, there's a lot of great science that goes on, but it's in Spanish. So it's therefore not accessible either. But there's so much data in so many countries, but because it's not in English or published in English, we're not getting hold of it. And so how are there ways that we can put this in so that all of this data becomes usable, especially for the countries that are producing this? And then for, these baselines that we're worried about, but also making sure that these baselines are relevant to the communities that we're working in. We have a lot of long-term research centers around the world. There's a bunch that are turning 100 years this year, 50, 60, 70 years old. Those are the baselines that we should actually be using, because that's what we need to be getting back to, not something from the 1990s. So how do we find this data, get it there, make it relevant to where we want to go, and getting these fish populations, in my case, or crustaceans or other fisheries, back to where they were. So we've been working on a few tech solutions along with everybody else. So the potential of remote sensing, and we've been really focusing on Google Earth Engine, open access, using Sentinel-2 imagery, another free resource, and sort of mapping mangroves at 10-meter resolution, and then creating dashboards that give people the access to that data, and so that they can interrogate it how they want and at different scales. Then there's the piece of environmental DNA, and we're working more with this in looking at how fisheries are recovering in general in marine protected areas or other effective conservation measures which local communities have enacted, but also in and mangrove restoration. So you can add this to whatever you're doing, ongoing practices, and it just adds another level. I think a lot of us know more and more about eDNA. And for us, it gives us an idea or a way to be able to scale and replicate, because you can leave these kits in the hands of communities and they can go back once a month. And you can also give it to 100 communities rather than having scientific expeditions that have to go out to all of these places, and you can only do it every once or every 2 years. So is this the way that we can get things moving? For us, we've found with fish structure, in some of our places, it's been really good to find out where the endangered species are. It's been nice for the communities to see the structure of their fish too. What are the commercial species of importance to them, where they are, and in what, relative abundances. So eDNA is a little bit tricky with some of that. But there's just these ways that it's more apparent to them and how they find these data interesting. But all of these do hit the EBVs that can then go up again to report, but also come back down again. And I think that this is, so some of the main caveats of all of this are we have to do the co-development. If we're moving forward with this biodiversity framework, what's the co-development for the questions on the ground with the communities that flip back up again, so that there's this conversation and movement of data up and down? Of course, we need the standardization and all of the metadata that was talked about in the previous session, especially with DSI, super important. For us as well, the curation of vouchers and libraries, this is a big job for the natural history museums. That's where our real taxonomic expertise come in, making sure that those vouchers are good for eDNA and other pieces so that we know that when those vouchers hit, or when those sequences hit those reference libraries, we're not going unknown fish 1, unknown fish 2. We're actually saying yellowtail snapper, red snapper, or whatever it is, because that's where the real data is. And then, giving a shout out to GBIF, OBIS, and GenBank, they do need money because the amount of data that we're about to produce on a weekly basis, monthly data is going to be insane, especially with some of this remote sensing and genetic pieces. So how do we make sure that that all remains open access, that everybody's happy with data sharing so that this information is freely shared across all of these different stakeholders? And then lastly, and this is thanks, Enrique, from our conversation last night, these technologies are moving super fast. And if they're not tied down to the essential biodiversity variables, we're going to have a constantly shifting baseline that we can't use data from 2 to 3 years ago because the technology has exceeded past where we want to use it. So how do we make sure that the technological solutions that we're creating are based on these essential biodiversity variables and that they will be for the long term and not just the short term fixes so that we can get some data now for the first round. So sorry if that was a bit quick, but that's me. Thank you.
Thank you, Steve. And I appreciate everyone sticking to their time until now. I know it's not easy. So next, I would like to ask Dr. Hilde Eggermann for her presentation. Hilde is a freshwater biologist by training with a PhD in global change research. She's the strategic coordinator of the Belgian Biodiversity Platform, a national science policy interface for biodiversity. She's also chair and coordinator of the European Biodiversity Partnership, Biodiversity Plus. She acts as Belgian focal point for IPBES, IUCN, for WCMC Europe, and her main expertise focuses on science-based policy support, research programming, and biodiversity policy. So Hilde, you have the floor.
Thank you, and many thanks to the organizers for having the opportunity to speak at this event and more specifically to explain the plans that we are currently having to set up this European regional observation network. I'm presenting this work on behalf of 2 organizations. So we have Europabon, coordinated by Enrique Pereira, who is also here with us, and then I myself representing Biodiversity Plus. And you will learn more about our organizations as I run through this presentation. So maybe first a little bit of the context for the biodiversity monitoring in Europe. It's actually quite clear in the sense that we have nature directives that are the cornerstone of the EU nature protection policy. So that means that member states know what to report to. We also have a new law upcoming on nature restoration. So that means that countries will also need to report on restoration indicators. But despite this quite clear legal framework, we do bump into several challenges to do the actual reporting. I'm sure that these challenges are not restricted to our own region, but we do lack a lot of data. There's gaps for certain taxa, habitat types, and ecosystems. The biodiversity monitoring protocols, methods, and indicators are not harmonized. In many cases, also, the reporting often constitutes a high burden because countries, they may not have the infrastructures or even the capacity to do the national integration of data that is needed to report back also to the European Environment Agency. Very often, we also have a confusing network of knowledge holders. We do not always know what is happening at the national level, subnational level, and how it relates to also some citizen science networks. So we need some kind of a mapping. And also, we see that the European IT infrastructure for biodiversity is not developed to a full capacity. Now, it's clear that not a single organization can overcome all these challenges. So that is why we are joining forces. So on the one hand, you have Europabon, whose main aim is currently to assess the feasibility of setting up a center to coordinate the monitoring activities across Europe. based on existing national and subnational biodiversity monitoring scheme. And to do this, Europabon is assessing the current biodiversity monitoring landscape to assess the gaps and the bottlenecks. And it's also demonstrating how the EBVs can provide this timely and relevant information for Europe, including policy relevant indicators to track progress towards the targets. So Europabon is producing outcomes, results, indicators, recommendations that need to be understood, but also taken up by the member states and the associated countries. So kind of moving beyond maybe a more technical exercise and creating the necessary capacity for those that are funding and steering the monitoring schemes in Europe, and that can also bring it down indeed to the local level. And these are often the ministries of environment and the environmental protection agencies, and also the ones that are in charge of reporting back to Europe and to the convention. And so that is where Biodiversity Plus is coming in. Biodiversity Plus is the European Biodiversity Partnership. It's composed of 74 ministries of research, ministries of environment, and agencies from 36 European and associated countries. And we're joining forces as part of the European Biodiversity Strategy 2030. So we have both the research policy actors on board, but also the environment policy actors. So that means that we can better bridge between science and policy, but that we can also embark on these biodiversity monitoring activities. It's not an entirely new partnership. It's building on an existing network that already existed since 2008, but it's more like an upgrade. It's co-designed with the European Commission, and it currently runs over a period of 7 years. Biodiversity itself has a broad portfolio of activities. The biodiversity monitoring component is only a part of it, but it's a very significant part. Another major component of Biodiversity Plus is more focusing on supporting and promoting research through annual calls. I'll come back to that later. But in any case, it's a perfect place to indeed have this link between research, monitoring, and policy. And it's very complementary to Europabon. It's the place indeed where the outcomes from Europabon can be operationalized. As we know, also Europabon is functioning as a regional bond. It's also one of the recognized bonds by Geobond, so it also complements the membership of Biodiversa. And of course, all this work, we also do it hand in hand with other organizations like the European Environment Agency, but also with important research infrastructures, just as GBIF. Now, coming back to Europabon, one of the activities that they have been doing is laying out a foundation for this European Observation Network. And they have been doing that by rolling out a user and policy survey to assess how we can improve biodiversity monitoring in Europe. So the report points to the gaps that I have referred to previously, but it also hints to possible solutions. And these solutions are multifold. Again, I think this is not restricted to Europe only, but it points to better coordination between these national and subnational biodiversity monitoring schemes, having more sustainable funding for biodiversity monitoring, also more harmonization of methods, protocols, and databases, more digitalization, more publication to GBIF, also the development and the deployment of new technologies and tools for biodiversity monitoring, better integration between what we have in situ and remote sensing data, stronger engagement of citizen science approaches, and also more capacity building amongst the countries. Europabon also worked on a catalog of monitoring activities, so more like some kind of a database that was then also used to map how biodiversity data collected in monitoring schemes across Europe flow through the different institutions and programs to produce these EBVs and other policy relevant indicators. So this also allows for a proper assessment of the gaps and the bottlenecks in the EBVs. And then last but not least, Europabon also is working on a series of policy relevant showcases illustrating these EBV workflows from observations to decision support. So now all these results are further being enriched and operationalized, as I mentioned, through Biodiversa. This means in different contexts and in different countries. So for example, more specifically, we are assessing to what extent these EBVs are known amongst those actors that are funding and steering the monitoring schemes, and to what extent they could be a good backbone also for setting up this European Biodiversity Monitoring Coordination Center. It's also the place to ensure ownership by environmental policy actors for such a center, to avoid that we set up a very top-down structure, but to really make sure that we create a structure that facilitates interaction between countries, that respects also some national and subnational governance structures, while at the same time indeed giving guidance and some level of coordination. Biodiversity Plus is also a good place to ensure best practices between countries. We do see that several countries have very good coordination structures in place, so definitely it's a place to showcase to other countries that can learn from that. And it's also the place to develop some guidance tools for harmonization of protocols, for methods, also for data interoperability. So then once we have these guidance documents, this can be rolled out by the center when we have it. It's also a place where we can have indeed this link with these research infrastructures. I already mentioned GBIF, and I'm sure we can come back to that later. And then also the work that we do, it's going all the way down to better use of monitoring data, both by researchers, but also by policymakers, both private and public. So it's quite encompassing. And then finally, and this is my last slide, in addition to the work that we do in terms of structuring the monitoring landscape, Biodiversity Plus is also, as I mentioned, supporting new research. And we have a call open currently on biodiversity monitoring. That call was launched in last September. It's a call of around 40 million euros, and we have 33 countries participating. So the topic is biodiversity monitoring, but it includes several sub-themes. One of these themes is also focusing on developing new tools and methods for collection and management of biodiversity monitoring data. So we look forward to work together with these normally around 40 pan-European research projects that will be selected from that call next year. So I'll conclude here. If you would like to know more about our organizations, I recommend you to just visit our website, to follow us on the social media. And in case there are ministries or agencies in this room that are not yet part of Biodiversity Plus and you are interested to join, please let us know. We are also open to non-European countries, and we are very happy to explain to you how you can join. So thanks again.
Thank you so much, Hilde and Ulrike. Great to hear about this partnership, and thanks to all the keynote speakers. I think we've heard about very important tools and programs, and also some of the challenges we still have. We have a few minutes now, so I would like to open it up for questions now for a few minutes. If you could say which organization you're from and your name first, and then ask your question. Alice?
Alice Hughes here on behalf of CBC-GDF. I work for the University of Hong Kong. Alex, fantastic work on using AI to uplift species. My question is partly on the need for what kind of data is required. A lot of the spatial data generated by BirdLife and IUCN, the spatial data in particular, has a lot of embedded assumptions on it. And one of the critical components of their approaches to listing species is the area of distribution, which, given that their maps are wrong, is often highly inaccurate. How are you getting around that in your approaches, or are you relying on primary distributional data through sources like GBIF in order to get a more nuanced and accurate understanding of species ranges? And do you also have minimum criteria that flag species that should just be data deficient? Because talking to some of the specialist groups from the IUCN, they will even say, well, we don't want to list too many species of data deficient, so we've given them a listing just so not everything is data deficient, which of course undermines conservation in the region. Thanks.
Thanks so much, Alice. I'll defer the question about how classification into data deficient species goes in the full red list assessment. But I think what we've been using is mostly spatial components. So to train neural networks, so in the machine learning approach, you need to have which variables you put into the model depends highly on how people have used that before. So it really learns from the data. Of course, there's no magic box. And when it comes to, for instance, things like fungi, where there have been so few assessments, and also many of those species that are triggered to the red list actually have not only be based on the spatial distribution of those species. So you really have the training data sets is the foremost criterion. We have not been used GBIF as such, but we've been doing a lot of data curation and cleaning because I think to a large extent, yes, it's usable, but we've also developed some automated methods, but then combine that with expert knowledge to create data sets of distribution, which then are used to feed into those models. I think there's a lot more that can be done. We just did a few examples, and there's been some other fast tracking of assessments using other forms of machine learning as well. In this case, I think we are confident that this is actually helpful, and because we can train and test, you can validate against part of your data, you can actually get some sort of confidence. But there's more that can be done, I think. Happy to.
Great, thank you for that. I saw another question back there. Can you get your way to a microphone there? Yeah.
So my question to the first speaker, to you, if let's say if a given region, you map the biodiversity and put an AI model for monitoring, so the kind of sentinel species that you use needs to be short life cycle species. Otherwise, you'll not be able to monitor the overall biodiversity. I was just wondering, have you thought of a few sentinel short life cycle species as sort of which you feed the data, real time data, so that you can continuously monitor using the AI model? Could you understand what I'm saying?
Sorry, could you speak a little louder? It was
Apologies.
very
Who's the
hard
question
to hear.
to? Is that just Steve? Or
Okay, I'll sort of speak louder.
Okay, sorry. Okay.
No. What I'm saying that let's say a given region, once you map the biodiversity and put into an AI model for a monitoring, the kind of sentinel species should be of short life cycle, so that you can continuously feed in real-time data for monitoring. What kind of sentinel species for a given region have you thought of, and how to sort of monitor? Because you need real-time data for monitoring.
Yes, I think that's a question to many of us here. It's a difficult one. It's of course much easier with trees or other static benthic organisms in the sea as well. But because species, many of them are very mobile, how do you actually do systematic conservation planning when things move around all the time? So I'm happy to be a botanist. So plants don't move that much. Some of them do, but at least it's a bit easier.
But the trees are very long life cycles, so you can't be indicators of loss of biodiversity, unless there is a
on the other hand, you can
destruction.
use remote sensing. You can satellite images to identify elephants and some other wildlife. So I think there's a combination there, and I think there's been some really interesting work done now on birds and GPS tracking of other organisms, and that's the kind of thing you would be able to fit into a model. Because if a species goes into an urban area or into an area which is being deforested, I think we saw, for instance, in Australia with the fires, was it about 3 billion animals that were killed by the fires? Of course, it has an effect on the how those species are doing in terms of their conservation status, but that usually takes many years until someone reassesses the conservation status of those species, and that's why the real-time approach, I think, is the one that we should be striving for, even if it's a bit difficult, and we still need some technological development to achieve that.
Okay, great, thank you. We we actually don't have, I'm sorry, any more time at this point, but we will have time for questions again later. But at this point, we're going to move to the second segment of the keynote presentations. And for that, I will turn it over to Gillian Campbell, who works for the UN Convention on Biological Diversity, where she is responsible for monitoring of the post-2020 Global Biodiversity Framework, including national action planning. So I couldn't think of anyone better for the moderator role. So I will turn it over to you, Gillian. Maybe we switch.
Good morning, everyone. So I won't take much of your time. I'm really just going to start with introducing the two presenters that are going to present, and then we'll continue with the panel discussion. I think most of you probably are familiar with the GeoBond. It's been referenced a number of times. Of course, this is the Group on Earth Observations Biodiversity Observation Network. And it's really working to coordinate the work across the collection of these EBVs and of biodiversity observation data at the ground level. And so the GeoBond has two co-chairs, which we have both of them here today. That's Maria Cecilia and Andy, and they are Maria Cecilia. I don't know how to say your last name, actually. Yes, Mdona. And Andy Gonzalez are going to walk us through how a global biodiversity observation system might be able to support the monitoring of biodiversity in the global biodiversity framework. And Maria Cecilia is, in addition to, of course, being the GeoBond co-chair, she works at the Humboldt Institute in Colombia, and Andy is with McGill here in Montreal. Okay, over to you.
Take you a while to
This is not. I don't understand.
That's weird.
Yeah, it is. Yeah.
Thank you, Gillian. Does everyone hear me?
Yeah.
Good. So I'm going to start with what I think GBIOS, this Global Biodiversity Observing System, needs to be able to do, how we need to transform our collective effort to provide the evidence, and guiding action for the Global Biodiversity Framework. And I'm going to start by pointing out that the Global Biodiversity Framework is a theory of change. It's about imagining where we want to be and reversing back, understanding how we can mobilize evidence, guide action according to the values, the knowledge, the repeated assessments of the state of nature to ultimately guide the adaptive decisions. And what we're talking about today here is, what is that minimum system we need? What is that information we need? Whether it's working from the bottom up with local communities, working from the top down with national governments, and everything in between. So one of the things I think is missing in the biodiversity conversation is a detection and attribution framework that our climate colleagues have been developing for decades. And I think we need to play catch up fast. Those of you who are familiar with some of the publications coming out of the IPCC know that they have had to do a lot of work to convince powers that be that humanity is responsible for modifying the climate. But when they do that, we're thinking about the entire Earth system. So we can see the climate system at the top here, we have the natural system down on the lower left, and we have the human system on the right. Those arrows are supposed to indicate how actions, for example, in the human system, through anthropogenic climate drivers like greenhouse gases, modify the climate system, which in turn may feed back to affect the natural system. What we're doing in this conference is thinking a lot about how everything is impinging on the natural system, how human actions or indirectly via climate are impinging on the natural system. But we're also thinking about how the natural system feeds back to climate and feeds back to human well-being, nature's contributions to people. So the current framing of detection and attribution is very much driven through the lens of climate. And I think we need to put the lens on the natural system and a principled process by which this global biodiversity observing system will support rapid detection and attribution that leads to action, not just detection. So what is detection? We have a forthcoming issue in Philosophical Transactions B, and in that we're supposed to clarify the conceptual landscape of applying detection attribution to biodiversity change. So detection of change, let me give you a definition. It's the process of demonstrating that biodiversity or an ecosystem process affected by biodiversity has changed in a well-defined statistical sense without providing a reason for that change. But we're providing a degree of confidence that we've detected change, and that change is commensurate with the goals that we are trying to shoot for, whether that is mitigating impacts or promoting actions that lead to restoration or recovery. Attribution, this is the process of evaluating the relative contributions of multiple causal factors to a change, either in a trend or in an event, like Alex was speaking to, a rapid climate change event. And that we always assign an estimate of a statistical confidence to that attribution process. Now, that might seem easy if we're just talking about, for example, deforestation, and we're trying to measure an indicator of change of forest extent. But in many cases, the causes of extinction or long-term changes in ecosystem processes involve interactions among multiple drivers happening at multiple scales of space and time. So the challenge is all the more difficult because of that. So when we talk about a future assessment of biodiversity change, I believe that we must establish the appropriate counterfactuals, meaning the reference states, the appropriate baselines, the appropriate references that account for the variability and uncertainties in the observations and the responses that we believe are causally driving outcomes, whether those outcomes are what we want to avoid or whether the outcomes we need. So let me boil all that down into a simple figure. So on the left-hand side, we have human drivers and actions, and they're interacting with the state of biodiversity, whether that's genetic, traits, populations, communities, and ecosystems, all the way up the hierarchical organization of life. And on the right are the state of natural and managed ecosystems. So what we want to be able to do is provide compelling, convincing, high-confidence statements about how action on human drivers is mitigating impacts on biodiversity and its state and rates of change. We want to be able to talk about impacts and attributions of how action on biodiversity impacts the functioning of those ecosystems and the services that they're providing to people wherever they are in the world. And of course, we want to be able to connect the dots. We want to be able to show that action on human drivers under the GBF are leading to appropriate outcomes in the middle on biodiversity and on the far right on the state of nature. So linking goals A, goal B, goal C, and all the way through in a principal system. Now, I don't really have time to go into this, but we have developed a framework where what's really important is the 5 boxes at the top. We know how to do observations. We've seen some compelling talks today about rapid observations, about using the best statistics to do estimation, and the best statistics to assign confidence in the trends we're detecting and the attribution to multiple causal drivers. What we are not doing right now, though, is guiding our observation process by the models that we think are important. The far left there, the causal models, which are establishing alternative understanding of the future and then using those expectations to drive effective monitoring. And all of that should be in a loop, iteratively improving our confidence that our action is leading to the outcomes that we want under the GBF. This is what Geobon is trying to promote as we move forward. EBVs are nested in that observation step. Indicators are being estimated. We're detecting on indicators. We're detecting on ABVs. And of course, we're doing strong causal foundational analysis on the far right. Now, this GBIOS, this Global Biodiversity Observing System, is country-led, but the idea is to stitch together the global mosaic of biodiversity observation networks into this robust observation, box 2, into robust detection and attribution, box 3, through to feeding that robust detection attribution into scenarios and forecasts, and on into policy decision, local action, and supporting management practices. We need to be getting this set up ASAP and doing this fast and doing it well and providing the support, the capacity to do this for every country, every party to the convention. So as you see here, that recipe I showed you in the previous slide is embedded in step 3 that every country will need to do as it tries to establish its NBSAP and the actions required to meet it. Now, let me give you just one quick example of what GBIOS will help us to do. What we want to be able to do is global detection attribution. That's just one element of what we can do. We can do it regionally, we can do it locally, of course. So here is those dots there are showing you an estimate of a trend at one place on the planet. It could be in the oceans, it could be coastlines, it could be on grasslands, wherever. And that cloud of data is something, the impression about the state of biodiversity for that trend around the world. If they're above the zero line, there's a tendency to be increasing. If they're below the zero line, they're tending to decrease. And across that x-axis is the degree of human impact. So expect this cloud to drop below zero as human impacts are increasing. But it's an uncertain process, and we're dealing with short time series, often with poor baselines, and we're incrementally increasing our understanding. But if you take that entire cloud, what you get is this distribution of our understanding of change. It has some uncertainty on top of it, but it's reflecting this global picture. In gray would be exactly what we think it is today as we embark on the post-global biodiversity framework. But what we want to be doing is bending the curve. And bending that curve away from the gray to the yellow requires systematically detecting an attribution of change so that we shift the distribution so it's above zero, so that we're seeing increases in the rates of recovery to bend that curve. And that is what we want GBIOS to be able to do, is systematically detect around the world that our efforts cumulatively across the planet are bending the curve and shifting the distribution of change through this detection attribution process. That was a lot of information. I rattled that off. Whew. This is my last slide. I would like us to be able to come back 10 years from now and be in a place where what we've done is we've responded to the challenge that our community is now able to move beyond research mode case studies and is developing monitoring systems and tools that can deliver rapid, reproducible assessments, maybe in the aftermath of notable ecological events, whether positive or negative, and that we are doing that on a scale of weeks to months, not years. We don't have time for lags and waiting. We have to have a responsive, actionable monitoring and policy hand in hand. So I will stop there and let's take this road together.
So how do I put this now? There. Okay. Well, thank you very much. So I'm very happy to be here today. And well, obviously, all of us have been working very hard to show the importance of monitoring. And I think we are moving in a good way forward as we have the monitoring framework being discussed, and that is a big achievement of recognizing the importance of monitoring. And I'm also happy to be today representing the Global South in this panel and being a woman. So that is also an important thing to have in mind here in my talk as you hear it. We have been hearing these 2 days of the importance of tracking progress and guiding action through good and enough monitoring and scientific basis. We have been hearing a lot of opportunities for new development in these fields that are already there, and we have to understand and use. And also, we have been talking a lot of about the gaps that they are already there. So, with that in your minds, I'm going to go into the matter, the monitoring framework that is being negotiated and discussed. For you who maybe don't know, there was a meeting in Bonn, an expert workshop on indicators, and they assessed a lot of indicators that were proposed in the process as being headline indicators. And they did a system of a number 1 to 5. 1 are indicators ready to go and use by all parties, and 5 are the ones that are not, or the least recommended for being headline indicators. And as you can see in the graph, 14% of those indicators are on the number 1. Now, that means that 86% of the indicators that have been suggested by the parties and observers as being headline indicators do not fill the characteristics of a headline indicator. So I also brought, I know many of you are not involved in detail to these negotiations, so maybe you are not aware of those characteristics that are needed for a headline indicator. So I brought some of the text that the negotiators are reading and discussing, and also those that are not in bracket. So, and highlighted in yellow, important words for you to pick up quickly. They need to guide, like, or talk to the overall scope of goals and targets. They need to guide planning and track progress. They need to be based on national data and calculated at the national level. They data and metadata need to be publicly available. Their methodology pre-review or validated for national use. They need to be regularly updated, and there needs to be a mechanism for maintaining the indicator in time. If parties and observers are the ones that are proposing these headline indicators, why do we have so few indicators that meet all these criteria? For 23 targets, we have like 8 indicators ready to go, and you are aware of all the whole dimensions of things that are in the targets. So some possible reasons that I want to bring to you today and think about maybe what we need from the countries, from the parties that are making the decisions there. So first, parties might be working in silos. So they might be not connected to all this really good work we have been seeing these 2 days. And they have been producing maybe their indicators on their own. Might happen that institutions working with indicators, both globally or regionally, nationally, do not make their data available. That might be also happening, and countries are not getting where the data is there, if it's there. Institutions do not publish in the pre-review journals their methodologies, and this was something mentioned by one of the speakers, Steve, I think. We do not publish in English, and many of the countries do not have this practice of using pre-review. Mainly global South countries do not do that on regular basis. Monitoring sampling might not be good enough for tracking progress, guiding planning, or keeping indicators updated. And that is very related to the last presentation of having in place this detection attribution system. Biodiversity indicators might not be developed thinking in the overall scope, and they are very specific. So when we review the list, it's like, this is not talking about the whole target or the whole goal we aim to track. Institutions are not able to sustain monitoring and indicator production. We need to enhance national institutions so that we can have this monitoring connected in a better way. So what do we need to do? Many things had come up now in the previous talks, but just 3 quick things that I think just put everything together. So one is this urgent need for global coordination of biodiversity monitoring. The other is that the achieving of goals and targets need this detection attribution system for guiding action. And well, countries need to have data and processing capacities in place to produce indicators with their data. So how can the scientific community be part of this solution? And well, we have been hearing a lot of options, and this is just one of those options. The idea of having a bone in a box, so a system. This is an idea of infrastructure, an informatic infrastructure. Let's say that can increase the capacity for biodiversity monitoring at the national level. Let's say I'm focusing this talk in the national level, And the idea of the bone in a box started about 6 years ago with some of the people that are here. Mike Gill, I know you're here. And we thought a lot about this bone in a box that was in version 1 a catalog of tools. Now it has been developed a little bit more, and now we have are working with the McGill QCBS Center and Humboldt Institute to develop this. So it has several blocks. One block is connect people through making monitoring projects more findable. The second block is to have the pipelines for calculating the essential variables. The third block is a methodology in place for optimizing that sampling to fill the gaps related to the indicators we need. And the block 4 is the pipelines for calculating the indicators, and these to be just connected with national and global data repositories and dashboards and indicators. So it's not creating a new dashboard, it's not creating a new repository, but trying to provide tools so countries can process their data, have the autonomy on their data, and develop and deliver the indicators. Now, this looks like that for now. So basically, is all this process that you need to calculate an indicator that you can put together in a pipeline from the data to the EVV and to the indicator, and you can run this. That way, through this, the academic community, the people developing the indicators can share their codes, and countries that need to replicate them can organize them here and run their pipeline. So this is one of the things we have been working. And also the block 3 on the designing and improving the sampling. We are focusing on the indicators for goal A and targets 1 to 7, so that we can talk about how to improve the sampling and have better indicators on those topics. So just general conclusions for our the discussion we're going to have in a few moments. The majority of the indicators will require the increase the capacities in the countries in 3 big areas. One is informatic infrastructure that will support transparency and reproducibility of the methodological ways to calculate the indicators. Then all the monitoring science, so supporting the filling of those gaps, better data, better models to better understanding what is happening, and strong institutions. We need to have stronger institutions at the country or regional levels, providing ownership and data sovereignty at the national level. That I think if we work the next years in these aspects for specific needs, we maybe in the next COP have a longer list of headline indicators on number 1. Thank you.
Thank you very much. I think that as opposed to going to questions, we will first introduce the rest of the panel and ask them perhaps to reflect on this question I think that you asked at the very end, which is, how do you think that we could build up the data systems that underpin many of these indicators, or the methodologies that underpin many of the indicators that are suggested, so that we really have operational indicators across these topics in the next few years. And so now I will, with that question in mind, introduce the panelists. And so I'll start with Tim Hirsch, who's come up here. I was like, where is he? He works with GBIF, which is the organization, the Global Biodiversity Information Facility, which coordinates a lot of this underlying observation information. And of course, they would play a key role in bringing this observation through their nodes up to the global level. And so I think that he's going to be in a really good position to tell us how he thinks that this can be developing over time. Then I'm going to go to Guido, who is Guido Saborio, who's there. And he is working in Costa Rica. And on the panel, I mean, as we've heard from all the presenters in up to this point, most of this work has to happen at the national level. And this is, of course, what's being said again and again in the meetings that we're having. And so as somebody who is doing this work in Costa Rica, it would be great to hear your views on how you think the system in Costa Rica could be further developed in order to produce additional indicators that could be used for monitoring and, filling some of these gaps in the near future. Similarly, now I will introduce Mike Gill. Mike Gill works for NatureServe, which is a network that operates primarily across Canada or North America. And he leads the work on the biodiversity indicators for NatureServe. And I think NatureServe has already done a lot of this work in terms of their own data, and now is also working to support other countries in developing similar biodiversity observation systems. And then I will go to Natasha. Natasha Ali has been working with UNEP WCMC for I'm not sure how long, but she has been working with me on the monitoring framework since the monitoring framework started its development. And so it would be great, Natasha, to also hear your views on what do you think that could be done to fill some of these data gaps. And I mean, I know that WCMC has been looking at specific opportunities and projects to work with countries. And so maybe you could also take us through some of those. So with that, I'll go in the order that I introduced everyone. So this will be Tim, then Guido, then Mike, then Natasha. Over to you, Tim.
Yes, thank you very much indeed, Gillian, and it's great to be taking part in this exciting discussion. So I guess my overall thought and message is when we're looking at the needs of what does a global biodiversity observing system require, the first thing to do is just work out what have you already got in terms of the components to be able to build that out and meet the needs, and what do you not have to create and of course, last of all, reinvent. And so I think particularly, I mean, where Andy was referring to, I guess it was the second box where you're talking about the basic observations, filling up that cube, so we have as much as we need of the basic information about where species are and what's changing in space and time. And look at, for example, you look at the GBIF map, we often see that, we know what the gaps and the biases are. So what do we do about that? We don't have to create something new, actually. We just have to try and direct those observations and that sharing of data to be able to fill as best we can the taxonomic and the geographic gaps that we know has arisen from a relatively opportunistic process of digitization, of encouraging data sharing. And this is where I think we can see the great synergy between the concept of an observing system and that vehicle, if you like, that GBIF provides to be able to bring together and to process those kind of observations. So how can the analysis of what needs to be detected, what needs to be observed, how can that actually go in and really help to fill the gaps and to create that raw content that's needed to make a global system like GBIF more representative and more useful to what we need. There's also, of course, when you share data according to common standards, there's often the complaint that, well, when you try and squeeze everything into the same model and you squeeze out all the good bits because you have to simplify and generalize the richness of the data that lies behind it. Of course, that has been true. And so what can we collectively do about that? There is a process going on at the moment in GBIF, a community-wide consultation to how we can diversify the model, taking account of new techniques such as eDNA, such as camera trapping, such as remote sensing, and how can we collectively create a better and more expressive way of sharing this very diverse very diverse sources of data in a way that can still be combined and shared into a global knowledge base. So we really invite everyone to take part in that and to look at collectively how we can have a better and a richer data model when we combine all of that into a single platform such as GBIF. I think it's critically important as, for example, the concept of biodiversity observation networks at the national level, at the thematic level, at the regional level, as these develop, how can they work closely together with existing networks of the data providers and those who have already engaged in the skills of data sharing of biodiversity informatics, for example, through GBIF nodes? How can they work together to make sure that those two processes of observation networks and GBIF nodes can really get to where we need to be? And then just a final point, I think, again, the concern that rightly exists of the idea of at the local level, the national level, putting data into a global system and then finding that you lose control, you lose a sense of ownership and attribution for what that data belongs to. Again, use the tools that are there. Encourage proper data citation so that when data shared using common standards, such as through GBIF, goes into a global system, makes it universally available for use, makes sure that that data is cited properly so that the contribution of those local systems is fully transparent and apparent in the onward use, so that those efforts of sharing data are fully recognized. And just, sorry, one final thing is, as part of that, we do need resources, we need capacity support. We in GBIF, through donors such as including the European Union, Government of Japan and others, have set up programs to build up communities of practice for data sharing and use in developing countries. We need much more of that to make sure that the opportunities and the benefits for standardized shared data are fully realized and recognized across all global regions. So I guess I'll stop there.
Thank you, Gillian. It is a pleasure to be here. So I guess that for Costa Rica, in terms of the monitoring framework, I think that we recognize the importance to adopt a monitoring framework. We think that is essential, so we are able to keep track of what we are doing and the progress that we are doing. And just going back to what Cecilia mentioned, realizing that by now we only have like 8 indicators that are supposedly ready to go. I think that that's something that we need to reflect and then to focus on how we can strengthen the different parties in their monitoring systems to be able to get that information. And also I think that it's really important that we see this as a process. So it is not just to adopting a series of indicators, it is adopting a process of how we can be further development the indicators that are still not ready, and to prioritize how we are going to be doing that process between now and the next COPs, and how we can get information or more parties being able to report in the 7th and 8th reports that are coming. So I think that's really important. If in the particular case of Costa Rica, so we have been developing a national program on monitoring ecological monitoring. We have been producing land use maps and using, getting into working with partners to use like a lot of technology to produce this information. And I think that our main challenge, and I think that is going to be for many parties, is how we can then translate that into the indicators that we are going to be using now. How we use the information that we are producing right now to fit into the methodologies or the requirements that are going to be related with the indicators. So I think that that's a big challenge. I think that it is possible. I mean, but I think that will require some thoughts. We will require the support of the academy, and to see how we can actually use that information to estimate indicators that we will need to use. And of course, I think that that's the case for most of the developing countries. We will need to have a lot of support in capacity building and resource mobilizations to be able to produce the information. That said, I think that we are really committed to adopt that monitoring framework and to move forward. It is really important for us. I think that that's key, and we should not leave this COP without having a monitoring framework. But I do think that we need to recognize that is a process, and then that we need to also decide how we are going to be doing that process through the train, so we don't lose that. We don't lose to keep track of the development of the indicators that are not ready yet, and the capacity building that the parties need. Thank you.
Yeah, thanks so much, and thanks to the presenters for really putting together a lot of the different ingredients that I think we need for a global biodiversity observation system that connects and scales from local to national, regional to global. Obviously, right now we're kind of in a chaos situation. I think of Alice Hughes's paper last year that looked at the inequities and availability of data globally. We did some analysis that looked at how parties used indicators in the 5th and 6th national reports. And the good news is that 11 times more frequently, those indicators are coming from national sources. The bad news is that it was chaos in that you had indicators coming using different methodologies, different standards, and we couldn't stitch it together. So what's the opportunity that we have now? I think we've got all the ingredients to put together a more coherent national and global monitoring and reporting system. And the trick here is, how do we do that in a way that we can quickly lower the threshold to stand up that federation of national bonds and reporting systems that are coherent and connect through data standards, through EBVs, and also through what I would call workflows or recipes. And the work we've been doing at the national scale in Latin America and the Caribbean, even up in the Arctic and Southeast Asia and Africa, shows that the opportunity here is really to take that monitoring framework and work at the national scale. So learn by doing, figure out how we can actually connect and integrate all these great technologies and solutions and methodologies into systematic tools and toolkits that talk to each other. So a lot of the focus of Nature Service Biodiversity Indicators program is to sort of connect and work together to find that way forward. A big focus for us is to really do a user-driven mapping exercise where we try and map out what are those core policy targets that are important to the nation. And the good news with that is that many of the core data ingredients and indicators can serve multiple policy targets, not just under the Global Biodiversity Framework, but UNCCD, national development plans, and so forth. So using that, we can really demystify that indicator production and reporting process. And I would argue strongly that we try and very quickly identify some practical, feasible core data ingredients that we can invest in and bring to bear many of the great and smart people in this room and others that can actually provide those solutions. The final thing I'll say is that I think there's an opportunity to increase the visibility of the innovations that are happening at the national scale within national agencies. We often don't really talk about some of the indicators that SAMBI, the South African National Biodiversity Institute, are deploying in different countries in Africa that use 4 data ingredients that produce a suite of indicators that connect to UNCIA, so natural capital accounting, to national development plans, and to many of the spatial targets. Those don't really make their way into the discussion on the headline indicators, for instance. And I think going forward, we need to sort of build that connected, integrated community where those innovations can find their way up and be shared around the world. And the final thing is that we talk a lot about the climate sector as our mentor or something that we can use as a solution. But the other one I would raise is actually the health sector, which seems a bit weird. But we've been looking at how health sector does upscaling. So how do they take innovations around rural health, things like that, and create the conditions or grease the wheels by which those can be quickly adopted around the world. And they have some nice, very simple, practical ways to do that through regional mentor models. So I'm thinking about not just GeoBond, but the Global Knowledge Center or Support Service Center as a way to sort of network and deploy and accelerate the transfer of technologies and best practices in different continents. How can we actually sort of create the conditions by which those different technologies can be synergized and upscaled around the world, and how do we actually embed those within institutions? There are ways to do it. As a biologist, we're not so good at that, I think, but I think we can learn from others. But again, I'll just say that I think we have all the ingredients. I think whatever we end up with the monitoring framework is a call to arms, is a user-driven framework which we can use and start to deploy at the national scale. Thanks.
That one? Okay. All right. Gillian said 3 minutes, so I'll do 3 minutes. So the first thing to note is that the role of UNEP WCMC is to come here and to support parties in the negotiation. So unfortunately, I haven't been part of this really important dialogue, and I haven't heard from many of the speakers. So if I'm saying things that you've already discussed, then please somebody just raise your hand and let me know. Then the other thing is just to highlight what the role of UNEP WCMC. So not only are we supporting the negotiations by being here in person and talking to parties, but we're providing the information around the indicators. So you may have seen that we maintain the website that holds the metadata for the indicators that are proposed and under discussion. And we create and generate various analyses on what is being provided to parties, or what is being proposed, for parties. And in doing so, we've got various sort of mechanisms and means for doing so through information documents to webinars and other ways. So in relation to the gaps, firstly, I think there's various ways of looking at this, and one is a sort of more of a wider thematic assessment of the gaps. And I know we're focusing on Goal A and targets 1 to 8 here, but of course, we need to think about the whole of the global biodiversity framework and all of the goals and targets. So one means of looking at the gaps is to look thematically and think, well, what are the goals and targets that aren't well covered by indicators, and should we be prioritizing there? A second means that we could be doing is the analyses that we've undertaken at UNEP WCMC, led by Samantha Hill, who has been looking at those indicators with spatial data. She's been looking at whether or not there are areas of the world that are not well covered by indicators, and whether or not there are thematic or other taxonomic gaps in those indicators. And we will publish that paper shortly. And then we'll think about whether or not support could be provided to indicator providers around that. In that regard, you asked me about projects. The Biodiversity Indicators Partnership, I think it was in 2016, we ran a Mind the Gap project, which is essentially for the Aichi targets. We provided financial support to biodiversity indicators partners to help them with developing indicators that met the gaps. We've got funding to do the same. So once we've got the post-2020 framework adopted here and we have a better idea of the monitoring framework, we will mobilise that funding to those partners to help them with either filling the gaps with those indicators that, with the goals and targets that don't have indicators, helping partners with developing indicators there, or, as we've been discussing here, filling the gaps in the existing indicators in which there are areas of the world or data that is missing. And when I come here and I hear these amazing talks, my mind's sort of blown as to where we would focus that very small amount of funding. So we really need to kind of keep in touch with the right people so that we can think about how we mobilise that funding and that support. The other areas of work that we'll be focusing once the post-2020 framework is adopted is, we're hearing parties say again and again that they want a streamlined set of headline indicators that they will use in national reporting. So we need to think about the indicators that are cross-cutting so that they can be used across multiple goals and targets. And we want to think about how we can support parties in using those indicators for multiple times to ease the burden on national reporting. Once in January 2023, through the GEF7 Early Action Support project, UNEP WCMC, UNDP and UNSD will go out to countries and start supporting them in their monitoring and their action plans, supporting national monitoring systems. And we will be doing capacity assessments with those countries to understand their needs for using national available data. And we will be then reaching out to biodiversity indicators partners to help us with that capacity building support. And then the final thing I'll note is that we're also, many of you have probably heard about this system that we are considering, which would consolidate many of the data sets and projects and networks that we've been hearing about in this room through a global knowledge support services for biodiversity. And the idea of that would be to take on board some of the really important science that is happening through GBIOS, through GBIF and other partners, and to try and to make that accessible to parties along with capacity building support. So that's where we got to. Thanks.
Thank you. So I wish we had more time. If we had more time, I think that we could definitely continue this discussion probably for the rest of the day. Unfortunately, we don't have more time. We are reaching the end of the session. In terms of concluding, I mean, if there are urgent questions, then please let me know. But in terms of my own wrap up, I did have some additional questions that I was considering. But what I think that we're hearing today, and also from many of the side events that I've attended throughout the week, is that there are a lot of the tools that we need in order to better collect data at the national level, to use new technologies to put ourselves in a better position in terms of monitoring the environment. Also, there's a strong interest from every party that I've heard from in terms of having better data. And there are also tools to take that data all the way through from data to statistics, to environmental economic accounts, to high-level indicators. We have a system. It's just that the system is all over the place, as was sort of said by Mike. You know, the system hasn't come together right now it's a bunch of systems. We have a bunch of systems instead of having a system, I suppose is the better way to say this. So the pieces are there, but it's that we as a community, as all of us, you know, from parties to NGOs to local level actors to international organizations, we have to find a better way to work together so that we're actually able to make this happen, so that we do have a fully operational system in the next few years. And so I think that's the real challenge that I will leave everyone with, and hopefully you can network with each other and try to brainstorm on how do we bring these different pieces together so that we don't remain in our own silos and we don't remain unconnected, but that we are all connected to each other. So I don't know if the organizers of this meeting wanted to conclude or if I'm concluding. So I'm concluding. Okay. All right. Well, everybody enjoy your lunch. Thank you.